wanted to make sure it's recorded. Hello, everybody. Hope you and your families are staying safe and healthy during this time. Welcome to the online version of Agilent's Seahorse Training and Advanced Techniques event. Under normal circumstances, this event would take place in your lab or at one of our Agilent sites. Please bear with us as we do the best we can to address this educational training virtually. I'm Rula, and I'm partnering with Kevin, who's your trainer and presenter, to be your host for today's session. We welcome your participation in our Q&A by submitting questions via the chat panel. As a reminder, this session is being recorded. And now it's my pleasure to introduce our presenter, Kevin Bittman. Kevin, please go ahead and begin.
Thanks, Rula.
Sure.
A couple of announcements. I am working out of my home office in Wallingford, Pennsylvania, which is a suburb of Philadelphia. My wife and my two daughters have lived here for the last 3 years, and I originally grew up in the Philadelphia area, just north of the city. So I consider myself a Philadelphian, and I was hired by Seahorse in 2008 as a field application scientist. And my role at the time was to assist the account managers with pre-sales presentations to help teach people about the instruments that we were selling and how they worked. I was in charge of conducting trainings for all of the users who had purchased the equipment or who were new to the equipment and requested trainings. And I also provide troubleshooting. So if people have some issues with data, they're stuck with something, or they just want to consult over what their data means, I'm one of the people that you can contact to achieve any one of those objectives. I am working out of my home office, and I just have a few announcements. I will be switching from screens, so there may be a delay sometimes when I switch from one screen to another. So just bear with me. I have 3 screens up right now, so I could quickly switch between different content. And there will be 2 breaks. We'll break after each of the first 2 lectures. And at the very end, there'll be a Q&A session. Let me back up a little bit. I will pause periodically to take questions, and I will tell you how to ask your questions in just a minute, but there will be frequent pauses for questions, and there'll be a Q&A session at the very end. Right now, what I'm trying to do is get my little laser pointer thingy working. And hopefully you are looking at my title screen and you can see the whole title and not my notes page.
That is correct, Kevin.
Excellent. All right, so the agenda, we are basically going to do a basic training. This is how we conduct a basic training. When we're in the field, we add a wet lab to this. So if you've ever been through this or you know somebody who has, usually your field application scientist will contact whoever the primary user is or the PI, and they'll discuss the goals of the training. In a wet lab training, we will talk about different cells that you can use with the instrument, what kinds of assays that you would like to run. We usually only do 1 assay because the assay itself takes about 2 hours to prep, and then we have about 2 hours worth of lectures and software demonstrations plus lunch. So typical training in the field would run about 4 to 6 hours. I'm going to do everything that I would normally do in the field for you. However, there won't be a wet lab. Instead, it'll just be a dry lab, and hopefully you'll feel comfortable getting in front of the instrument and giving it a go. You're always welcome to contact me with questions. If you are going to run an assay, you're welcome to contact me for a review. We've even done tutorials right in front of the instruments if necessary. I'm not alone. I have several colleagues that can assist as well. My primary territory is the northeastern United States and eastern Canada. So I cover everything from about Philadelphia, Wilmington area, all the way up to Quebec City, and as far west, if you will, as western Pennsylvania. So everything in that seaboard corridor are the accounts that I would normally interact with, and my colleagues would cover the southeast and the Midwest and the west. So if you're tuning in from, say, Arizona, I know who your field application scientist is. Welcome to contact me, and I'll put you in touch with that person. So we're going to do an introduction on what the instrument does and how it does it, and then we'll talk about the actual steps you would need to perform in a lab. Now for the steps, there are 3 different Seahorse instruments. So there are different steps for each of the instruments. I will do the best I can to cover them, but don't hesitate to ask questions. And on the next slide, I'll show you how to ask questions. And then we're going to get into data analysis. I want you all to know that we released last month an online version of our software that is Apple compatible. One of the biggest questions, probably number 4, one of the top 5 questions I get is your software compatible with the Mac? The default Wave software is a PC downloadable for free piece of software, which I will show you today. However, we recently released an online version that just requires a browser. It works best with Safari and Chrome, and we'll get into that. And then we'll wrap up with Q&A towards the end. I budgeted about just about 3 hours. That's how long it usually takes to do these steps, and we might finish sooner because of the format. We're not walking from one place to another, and I'm not sure what to expect with questions, but we'll see how it goes. So hopefully we'll wrap up around 1 o'clock and I won't starve you all. I remember once years ago, and I trained Jay Dunn, so I know some of you know him. He started about 2 years after me, and Jay was doing a training once and his watch broke. So he was doing his lectures and his watch kept telling him it was 11:30. So he was thinking, oh my gosh, I got another half an hour to talk. So he kept trying to find content for people. And then sometime around 12:30, somebody raised their hand and said they were really hungry. Could we break for lunch, please? And he's like, well, but we still have a half an hour. And then they went into the room at the back, clock at the back of the room and found out that, well, it was actually 12:30. So I don't want to starve people. All right, so if you do have questions, because it's a large group right now, it's hard for you all to interact and ask your questions audibly. So what I'd like you to do is chat your questions to the host. That's Rula Barakat. Rula and I worked together for many years in New England. She was the account manager, and I was the field application scientist. Is now the Eastern District account manager, district manager. So there's a handful of us that report to Rula, and she helps us out and make sure that things like this go smoothly. And she is here to moderate today and also to take your questions. So if you ever have a question, just use the chat feature within the WebEx and chat your question to Rula. All right, so let's get started. Thank you very much for attending. I really appreciate this. It's very nice to see that there's so much interest, and it's very humbling, and I'm very proud. So thank you very much for joining us. What I want to do is get started by talking about the Seahorse instruments. The Seahorse instruments were designed to measure the 2 main energy-producing pathways, notably glycolysis and mitochondrial respiration. And we'll get into how it does that in just a few minutes. But here's a picture of the 3 basic instruments that we have available for you to use. There's the XFe24, the E96, and we have something called the HS Mini. It's also called the XFP. Going forward, we will refer to it as the HS Mini. They differ in the number of wells that you can sample as well as cost. The XFe24 has 24 wells, the 96 has 96 wells, and the HS Mini has 8 wells. They are purposed differently. The XF Mini is a high sensitivity instrument. You can use fewer cells with that instrument than you can with the other 2. It's ideal for working with small sample sizes, human samples, rare cells like T regulatory cells, and that's essentially what it was designed for. The XFe96 is your higher throughput instrument. With that instrument, in my hands, I recommend running up to 24 different groups at a time. So it's a high throughput respirometer in that regard. It uses less material per well than, say, the XF24, but more than the HS Mini. So this winds up being one of the more popular of the 3 instruments. The XFe24 was built off of our original format, the XF24. That was our first instrument made. These instruments are ideal if you need extra space in the wells, larger surface area. They work with tissue and white fat. You can run tissue in the HS Mini as well as the XFe96. White fat, however, does float, and we have a specialized screen that can be used with the XFe94s if you wanted to work with a chunk of, say, subcutaneous fat. We have a number of kits that help you with your research and guide you through getting high quality data. And I will be featuring many of these kits through this opening presentation. And the magic in the instrument is these consumable pieces called cartridges, which we're going to get to know pretty intimately in a couple of minutes. So I mentioned that the instrument measures the two main energy producing pathways, glycolysis and mitochondrial respiration. Glycolysis is the metabolic breakdown of glucose into pyruvate. The glucose is enzymatically converted to pyruvate that produces 2 units of ATP. For glycolysis to continue, the pyruvate will be fermented into lactate. This recycles a proton that can be used as a reducing equivalent for glycolysis to continue. Lactate will be extruded from the cell and it will carry 2 protons along with it. So when a cell is glycolytic, it will start to acidify the extracellular media. So common ways to study glycolysis in cells in culture would be to look at the acid content of the media or to measure lactate. And you're probably familiar with both avenues of exploration. In the Seahorse instrument, the rate at which the protons get produced can be converted to a pH and measured in millipH per minute, a rate, which we call extracellular acidification rate. Of course, if you just want to measure the protons, this is going to be on a linear scale, unlike measuring pH, which is a log scale. You can express your data as proton efflux rate, which will be in picomoles per minute. It's this metric that we use if we want to look at glycolytic rate as well as ATP, because we can take advantage of the relationship between protons and ATP production. You probably know that glycolysis produces 2 ATP units and 2 protons. So if we measure the protons, we can directly convert those into ATP units. The other measurement that the instrument makes is on mitochondrial respiration. And there what we're measuring is the oxygen content. Mitochondria will produce a lot more ATP than glycolysis. Up to 36 units of ATP per molecule of glucose can be produced with mitochondria. But this requires the production of a membrane potential driven by protons. So the intermitochondrial membrane will form a proton dam or proton gradient. If you've got a proton, you've got an electron, and electrons are dangerous. They will damage things. So oxygen is required to reduce that electron to water, and that is the process of respiration. So if you don't have oxygen present, the cell tends to favor glycolysis. And there are some circumstances where a cell may favor glycolysis, even when there's plenty of oxygen around. And this was a curiosity back in the 30s by Otto Warburg when he studied breast cancer cells. Breast cancer cells are glycolytic even if you pump them full of oxygen. And at the time, the thinking was, well, why would a cell choose the less efficient energy producing pathway? And it became clear that cells may not be doing these pathways just for the energy. There are many steps and many products as a result of these pathways, and the cells might be interested in one of those products. Glycolysis, for example, can feed into the pentose phosphate pathway and make nucleotides and NADPH and lipids. The cells that need to divide, such as an activated immune cell, like a dendritic cell or a neutrophil, might favor glycolysis so that they can start to expand. And that would require more biomass, and glycolysis is favored for that. So we're going to be measuring oxygen rates of consumption as picomoles per minute, oxygen consumption rate, and either extracellular acidification rate, millipH per minute, or proton efflux rate in picomoles per minute. So how does the instrument do this? The instrument takes advantage of a piece of plastic that we call a cartridge. The cartridge here looks kind of like a comb. Each one of these bristles is actually a sleeve for a fiber optic light pipe to insert into.
If you don't mind muting yourselves, please. There's some background noise. Thank you. All right, go ahead, Kevin.
So these sleeves here are guides for a fiber optic light pipe that's inside of the instrument. During an assay, the fiber optic light pipes will insert into that sleeve, and on the other side of the sleeve is a spot that is a piece of plastic that's embedded with fluorophores that react with oxygen and fluorophores that react with protons. And the reactions are quenching. During a measurement, your cells will be adhered to the bottom of your wells. This is somewhat of a requirement. The material doesn't necessarily need to be stuck well to the bottom, but it should be resting on the bottom. The reason why I say it that way is that people have successfully run bacteria, C. elegans, zebrafish embryos, and in some cases they simply place them on the bottom and let them move around. As long as they don't all go to the sides of the wells, you'll get very good data. If you're working with cells, they'll stick to the bottom. If you're working with immune cells, which are usually suspension cells, we'll spin them to the bottom of the plate to make sure that they stay at the bottom of the plate. The reason why the material needs to be to the bottom is after the fiber optics engage the sleeve, they're going to push down to form a microchamber. There'll be about 200 microns from the bottom of the well to where the spot is. And we're going to sample the amount of oxygen and protons in this microchamber, which in the XF24 is 7 microliters, and in the XF Mini or the HS Mini and the XFe96, it's about 2.3 microliters. So it's a very small volume. And it's a small volume because we can pick up changes and gradients very quickly that way. The measurement itself only lasts about 3 minutes. Now remember, the fluorophores react with protons or oxygen, 2 different channels, and they're quenching. So when the cells consume oxygen, the fluorescence is going to go up. And we can measure that fluorescence and then convert it to an oxygen tension, measure that oxygen tension over time, and convert it to a rate. So the amount of time that was required to make a measurement is 2 minutes. Most of the time we set this to 3 minutes. So we're going to do a 3-minute measurement, and then the probes are going to raise away, and then they plunger back and forth. The plunging motion is to make sure that anything that you've added to the wells is well mixed, and also to mix oxygen and buffer back to the surface of the cell. This ensures that the cells don't become hypoxic or too acidic. So on the screen right now, what you're looking at is what we call the level data. You see on one axis the level of oxygen, on the other axis you see time. And during each 3-minute measurement, the instrument will sample an oxygen tension point every 12 seconds. So in a 3-minute measurement, you get about 14 data points to make up your slope. The instrument will then calculate the slopes of these curves and return for you the rates of each of them. On this particular data set, all you're looking at is 2 different cell densities of iPSC cells. So the blue is 60,000 cells per well, and the red is 100,000 cells per well. When we convert the data to rates, it looks like this. Now the software, the Wave and the Seahorse Analytics will automatically give you the rate data. If you want to look at the level data, you can certainly do that just by switching over to level. In my work, I always look at the level data because when you send me data to look at, I want to make sure you're within a tolerable range for the instrument and that you didn't forget to double check pH. So pH is another form of level. So that's one of the troubleshooting things that I do when I look at your data. So what could go wrong there? Well, if your media has evaporated, your oxygen tension is going to be way off. So if you noticed in the oxygen data here, we're starting at about 148. If you run out of oxygen, then this is going to be way down here. If you get an air bubble in there, it's going to be way up there. So I'll look to check to make sure there wasn't any evaporation or issues with volume. And as far as pH is concerned, this axis could be pH, and you want it to be sitting right around 7.4. So I mentioned a tolerable range. The best range to work in is between 100 and 200 picomoles per minute for each of the instruments. The XFe24 can go a little higher than the E96, about 800 picomoles per minute. The XFe96 and the HS Mini will start to bottom out as you'll run out of oxygen up around 600 picomoles per minute. So I usually recommend that people try to adjust their cell density so your respiratory rates land somewhere between about 40 and 200 picomoles per minute. That's a pretty good range. And you'll notice the different cell types will require different cell numbers. And I'll get into where to find some information about that later. But as a general guide, if you're working with an adherent cell line, we usually run 10 to about 50,000 of those. For example, C2C12 cells, a muscle myocyte line, you only need about 12,500 cells per well in an XFe96 and about 25,000 in an XFe24. If you're working with T cells, you need many, many more cells, about 200,000 in the XFe96 and maybe 400,000 to 500,000 in the XFe24. So I'll break here before I get into the injections and ask if there are any questions.
Kevin, so far we don't have questions. You can go ahead and go on. Maybe people are still thinking about it.
Okay. So the instrument, in addition to just making these basal measurements, also has the ability to inject compounds. In the very first assays that were run with the instrument, toxins were put in there. The first publications were from toxin safety groups, where various compounds like the biguanides and the other group, the statins, were just placed in the ports, and then they were injected to see if it had any bearing on respiration or on extracellular acidification. And interestingly, it did. Drugs like fenformin caused increases in extracellular acidification, which could lead to lactic acidosis. There are 4 ports in the instrument, and they are built into the cartridge. So you would load whatever drug you need into the cartridge, and the volumes, this is probably the part that you will have to learn, maybe the hardest part to remember, is each instrument has its own volume. So for the XFe96, typically you load between 20 and 30 microliters into each of the ports. The same for the HS Mini or the XFP. For the XFe24, the volumes range from 50 to about 95 microliters in the ports. As far as the plates, they are different with different surface areas, which I forgot to mention. The XFe24 has a surface area of 0.32 centimeters square, which is the same as a standard XF96 plate that you might purchase from Falcon. The volumes, however, are a bit larger because they bevel the wells to accommodate displacement that would occur when the fiber optic light pipes go into the wells. So the volume for an XFe96 is between 150 and 200 microliters, and we recommend starting your assays at 180 microliters. And that's the same for the HS Mini. For the XFe24, we recommend starting at 500 microliters, and the volume goes up to about 2 mils. So we have protocols and publications that will walk you through how to manage the media and the injector ports. You must load the injector port.
Kevin, my apologies. I do need to interject. I did get 4 questions as soon as we moved on, so you let me know when you're ready to take them, okay?
Yeah, sure. Now it's fine.
Okay. So the first question, and it sounds like everybody typed them at once, which is a good thing. One question that came in is C. elegans do not attach. Could you please comment on assessing C. elegans which do not stick to the bottom? Still published data with the XFe96 out there. Could you please repeat?
Is this all the same question?
So I'm not sure. It's a follow-up question. It says, please repeat the recommended OCAR range.
Oh, okay, sure. For C. elegans, there is a methods paper out there from Johan Arowitz's group that I will pull, and I'll try to create some content for everybody who attended today. I forget what journal it's in, but he has 2 publications with C. elegans. There's a couple publications that came out of Joel Meyer's group at Duke, and I've worked with a fellow named Ken Norman at Albany and Dr. Davidson, but I can't remember her first name, at Princeton. You're right, the C. elegans don't stick. What we found out is that when we gently place them in the center on the bottom of the wells, they wiggle around, but they don't really move. And what we do is we adjust the measurement parameters within the instrument. We put a wait command in between the measurements to allow the C. elegans to settle again. So we'll do a measurement, then it'll go through its mix phase, and then we'll put like a 2 to 5 minute wait to allow the C. elegans to basically just redistribute themselves. You will get a bit higher variability with C. elegans than you would, say, a cell line, but we've gotten some very nice data working with C. elegans. As far as the dynamic range for the OCAR, the listed dynamic range, if you were to go online, is going to tell you it's between 20 picomoles per minute and about 1,000 picomoles per minute. However, in experience, I recommend trying to keep your rates below 600 picomoles per minute because you can run out of oxygen in some circumstances within the well. So I always recommend adjusting your cell density so your basal rates of oxygen consumed fall somewhere between 40 and about 200 picomoles per minute, regardless of what instrument you're using.
Okay. One more question. Can we run it under hypoxia conditions?
We do have a hypoxic protocol, which is on our website. Our protocol assumes you have a hypoxic chamber. So you would place the whole Seahorse instrument into a hypoxic chamber and then run your assays. And we validated it down to 3% oxygen.
Okay. So I will address this question, Kevin. Will this recording be available later? I don't have that answer for you right now, but I'm working to have it available for you. We are recording it. It just has to go through several processes. However, I will send you my email and Kevin's email. We can set something up as a follow-up for you.
And I encourage everybody to ask questions because we will have a nice dialogue, a nice Word document of all the questions. So we can put together content to send to everybody based off of your questions.
Hi, Kevin. Hi. Can I ask one question? The cells which I work with, they are the muscle stem cells, and we isolate them. They are the primary isolated cells, and they don't stick easily to any surface or any plate. So would you recommend us to use something like a cell tag?
Yeah, I usually cycle through different adherent molecules. I start with whatever you know has been published. For example, with muscle myocytes, and this may not be true for your particular line, but with muscle myocytes, people tend to use laminin and fibronectin. So I would recommend that first. If that didn't work, then I would go to poly-D-lysine. If that didn't work, then I would recommend CellTac. And if that didn't work, then I'd recommend Matrixol. So I kind of go through that cycle.
Okay. Mm-hmm. And cell tag doesn't have any adverse effect on the cells, right?
Correct. It does not. In order to, I'm happy to take audible questions, but to make it a little easier because it's a large group, like I said at the beginning, I'm very flattered because I didn't have so many people to join. But when we start to exceed about 20 or 30 people, it gets hard to do questions with the audio.
Yeah,
so,
and building on that, Kevin, I'm taking all questions down. So if not everything gets addressed today, we'll follow up with an email for sure.
Okay, thank you.
Oh, you're welcome. My pleasure. Nice to hear from you. The instrument has injector ports, so you can do a basal measurement, inject a toxin, or inject a particular compound that may tell you more information about glycolysis, glucose, or respiration, such as oligomycin. And the way this works is that you would load drugs into the ports manually with a pipetter, and the instrument will be instructed by you to inject at a certain point in time. The injections are supposed to be complete. So when you tell them to inject port A, one of the 4 ports, all the A ports should blast their contents at the same time, and everything that's in the should go at the same time. So I'm going to try to show you this movie here, see if it works, which illustrates the injection and measurement system. So the instrument will form a seal, and this allows for air to pass through a port and inject the contents. Notice how all the ports go simultaneously for port A, and now it will mix and measure, and then it will repeat the process for the second port. So in this particular example, there were 2 injections. I'll get into the specifics of loading the ports in more detail in a little bit. And let me get my laser pointer back. So hopefully the movie works. Sometimes over internet things get kind of choppy. All right, so that's how the instrument is designed and how it works. I didn't go into any of the history, and if you read the abstract I sent out, I promised stories and props. I've already started telling a few stories. Unfortunately, I can't use my video for the same reason why we asked about the audio. There's too many people, so we had to shut off the video. I do have some tools here that are fun. When I first started at Seahorse, they gave me a little piece of the fiber optics. I have that. I've got some cartridges. I've even got an injection manifold. So I was hoping to show you all that, but I've got some pictures I'm going to show you guys after I finish the first half of this presentation. The instrument, where did it get its name? I like to tell this story. So it's called the Seahorse instrument, and now it's called the Agilent Seahorse instrument. The instrument was designed by an engineer named Andy Nielsen and his friend Jay Teach. Jay Teach went on to be the CEO of Seahorse, and Andy Nielsen was the chief engineering officer. Andy is still with Agilent, and he designs all of our new products. Jay retired. He owns a horse ranch, and he's become a consultant. I think he lives in New Hampshire. Very, very nice man. When they got together and they built the instrument, the first version of the instrument didn't work terribly well. They used thermal imaging rather than fluorescent imaging, and they called the company Thermalgenic Imaging. When they switched over to the fluorescent imaging, they decided, well, we need a name change. And the VP of sales at the time, a fellow named Steve Chomitz, said, you know, you can't name your company after what it does because it's going to be hard to find. We need to come up with a noun, something creative. So they held a contest, and Jay decided he wanted to name the company after a racehorse. So he kept recommending racehorse names, and they actually had a hat with names in it, and they put names in there, and people kept pulling off names of racehorses. Well, Steve wanted to name the company after a beach, so he put names of beaches in the hat, and they kept coming out with a beach name. So they've got this combination of beach and horse names, and then somebody in the room said, why don't we just call the instrument the Seahorse instrument? And that's how the instrument got its name. So what can you do with a Seahorse instrument? This diagram here walks you through a suggested workflow for getting closer and closer to mechanism of action when you're studying mitochondria and glycolysis. It starts, and these are all kits that have been designed, validated, and published. So these are backed by your peer reviews. And I like these first two because they are phenotypic in essence. In fact, this one we call the cell energy phenotype test. It lets you know if your cells are glycolytic or oxidative, and it can do this across different models. So maybe you've got a muscle cell, and then you've got a muscle cell and high glucose. You can compare the two and see if the glucose has any bearing on oxidative phosphorylation or glycolysis. The other assay, which is a great start, and this is the one that ships with the instruments, is the ATP rate assay. This assay is phenomenal. It's a 2-injection assay, very easy to use, and it lets you know how much ATP your cells are producing. But unlike a molecular marker assay, like a luciferase assay, it doesn't tell you the total amount of ATP made. It tells you the rate at which ATP is made, and it tells you the rate at which ATP is being made from both glycolysis and oxidative phosphorylation. This has several advantages over a luciferase assay. One, the rate is sensitive to supply and demand. A cell is going to try to maintain its ATP levels. So if it encounters a stressor, one of the two pathways is likely going to be upregulated to maintain ATP. If you run a luciferase assay, you might not notice that because the ATP content, the total amount that's sitting around in the cell, doesn't change. However, if you look at the rate of production, that would change. As supply goes up, the rate of production also goes up. And then the second advantage is it tells you which pathway contributed to the change in rate. I really like this assay, and I think these are great starting points to get a feel for what your cells are natively doing and how they might respond to stress. Then as you drill down, we have specific assays that answer more specific questions. This assay is a cell mito stress test, and we have a kit for this as well. And this assay lets you know about mitochondrial health and fitness. Then we've got a substrate oxidation assay, which is one step further towards mechanism of action, in which we assess mitochondrial function in the presence of inhibitors of either glucose oxidation, glutamine oxidation, or fatty acid oxidation. So this will let me know what particular substrate the cell prefers to oxidize. Then we've got the glycolytic rate assay, which is specific to glycolysis. So if you're interested in something like the Warburg effect and you want to see if you can adjust, or actually viruses, since viruses cause changes in glycolysis, this is an excellent assay to run to look at glycolytic changes in your cell. If GLUT1 is upregulated, you're going to see a change here, but these changes occur pretty quickly. It's also a great assay to run in combination with your activation assays to look at T cell, neutrophil, or dendritic cell activation. And we have another assay called the glycolysis stress test, which looks at glucose utilization by the glycolytic pathways. This assay requires a little bit of starvation, so we usually recommend the glycolytic rate assay. And then further down, we've got assays that are specific to mitochondria. In fact, isolated mitochondria and permeabilized cell allow you to test each one of the mitochondrial complexes to see if one of them is damaged or changed in your model. For example, Parkinson's has a known complex 1 deficiency. So if you have done something to try to change the makeup of complex 1, this would be a great assay to assess whether or not your protocol has worked. So let's start by thinking about ways of looking at data and how these kits can help you do that. First, I just want to cover basal rates. Basal rates are helpful. Don't feel that you've got to run these long assays. This can be a very quick and easy to run assay to get you data very quickly to determine what to do next. On the screen, you're looking at a histogram. We call it a phenogram or the XF energy map. All we've done is we've taken the basal rates for 4 different cell types and plotted the OCAR on one axis and the ECAR on the other. And this is a cartoon, so it's not real data from real cells, but it is correct in the orientation. For example, if you were to seed 40,000 neurons on an XFe96, you would get an oxygen consumption rate of around 100 picomoles per minute. If you were to seed 200,000 memory T cells on an XFe96, you would get about 50 picomoles per minute of oxygen consumed by those cells. So you can create a map like this and compare across different cell types and across different conditions. So let's ignore this for a minute. In this condition here, you're looking at a kidney cell basal rate, and then in another well, a kidney cell in which fumarate hydratase has been knocked out. So you compare a wild type to a knockout. And in this condition here, which is great for bioprocessing, we're looking at passage 12 versus passage 36 of C2C12 cells. And this data was drawn, both of them were drawn on real world experiences. This was done in a group that studies obesity, and they noticed that if they passaged their cells for too long, they became very glycolytic and they no longer responded to fat. And this was from a group at NIH in collaboration with a group at Harvard looking at renal cancer. So let me go back to this other one that I said to skip. This one here is called the cell energy phenotype test. In this particular assay, we go one step beyond basal. We've got our basal rate here. Then we do an injection of a stressor. The stressor is a mixture of oligomycin and FCCP, which drives glycolysis and oxidative phosphorylation. You'll learn why in a few minutes. The second measurement is just what direction did that carry the cell into? If I force the cell by stressing it to make ATP, is it more oxidative, is it more glycolytic, or is it a little bit of both? Neurons are very oxidative, ton of mitochondria. So when we stress them, you see this increase in respiration more so than an increase in glycolysis. So that is your energy map and your cell energy phenotype test. And here are some representative examples of that. So on the left-hand side is the actual kidney data that I had mentioned. This was from Marston Linehan's group. He's a surgeon at NIH. He's collected some samples. They've run in their labs, and over a period of time, they'd identified that there was a mutation, I think called the Van Hippel-Lindau region, which is somehow related to coding for fumarate hydratase. So they took normal human kidney cells and they knocked out fumarate hydratase to create a model of renal cancer. And you know, when you do this kind of work, it doesn't always bear fruit. The cells don't always carry the infection, or they don't have the mutation. So you grow up lots of clones of them. And what they're done is they've sampled the clones functionally using the Seahorse. So this winds up being target validation. And they show that clone number 1 and clone number 19 show that there is a reduction in oxidative phosphorylation, which is what you would expect if fumarate hydratase, which is in the Krebs cycle, is no longer present. In the middle here is a group that was interested in studying lymphoma. And one of the things that they noted is that the lymphoma was sensitive to TRAIL, tumor necrosis factor binding ligand. They could kill it, but only under certain circumstances. For example, if glycolysis is inhibited. But there isn't very many good treatments out there that block glycolysis. There's one that's coming out from a new company. I think it's called WP122, which is being researched to study coronavirus. But otherwise, 2DG by itself doesn't do a very good job of getting into cells. So this group was trying to find another sort of therapeutic. So one thought they had is, well, if glucose is important for this particular response, what happens if we just remove glucose? And to their surprise, when they pulled glucose, the tumor necrosis factor binding ligand, TRAIL, no longer killed the cells, which was unusual, unexpected rather. So they run a Seahorse assay and they demonstrate that when they pull glucose from lymphoma cells, the cells become much more oxidative. And this somehow enabled them to circumvent the treatment, maybe because it was no longer pro-inflammatory. I don't know for sure, but the finding was that the cells found a way around. I call it the Jurassic Park model. You know, you put a bunch of dinosaurs on an island and they're all male dinosaurs, and somehow they start to propagate anyway. On the far right here is an example of beiging of white fat, or at least an attempt to beige white fat. The one technique to try to manage obesity is to pump mitochondria into white fat to make them burn more fuel, especially fat. In this case, what they did is they tried to change the fate of the white fat by treating it with sonic hedgehog. If you get success with this type of technique, the tissue will beige in color and will become more oxidative. In this example, it actually became less oxidative. The sonic hedgehog likely deprogrammed the fat and made it more glycolytic, which was the opposite of what they wanted. So the technique didn't exactly work. However, it gave them some pretty interesting information that they could use for future research. So that is just looking at basal rates in these three cases. Now, the cell mito stress test is one of our flagship assays, and we have kits and plenty of resources to help you with this specific assay. This assay now, we're looking at oxygen consumption on one axis and time on the other. And what we're doing is we're injecting in sequence 3 different compounds, oligomycin, FCCP, and rotenone mixed with antimycin. The oligomycin is an ATP synthase inhibitor, which will cause a decrease in respiration associated with the amount of ATP being made. So the bigger this drop, the more ATP your cells are making. In fact, in the ATP rate assay, we take advantage of this to calculate ATP units. The next step is to add the drug FCCP. FCCP is an uncoupler. It forces mitochondria to respire, but without the added benefit of making ATP. So you get maximal respiration with FCCP. This drug is the only drug in our kits that will need to be titrated. And it should be titrated against every cell type and every cell line. And that's because of how it works. FCCP is a proton ionophore, and it will carry protons down their gradients. And I mentioned earlier that the mitochondria have intermitochondrial membrane potential driven by a proton gradient. That's what FCCP attacks. It will completely destroy that. If you add too much FCCP, you will literally blow up the mitochondria. And if that happens, then you will get no respiration. So too much FCCP, bad. Too little FCCP, and you won't see a maximal response. So you got to find that sweet spot. And the sweet spot is usually between 250 nanomolar and 2 micromolar, depending on the cell type. So one of your first experiments, if you run this assay, will be to titrate your FCCP. We then finish this with antimycin A and rotenone mixed together, which blocks all mitochondrial respiration. What's left over is non-mitochondrial, and what the assay reveals for us is basal respiration, ATP coupled, spare capacity, which is the difference between your max and your basal. Spare capacity is a huge asset to this experiment. A lot of different conditions will affect that spare capacity. In terms of how to interpret your data, oftentimes the spare capacity is linked to mitochondrial mass or substrate availability. So if you see changes in spare capacity, ask yourself, was my treatment done overnight or was my treatment done for a few minutes? If the treatment was done for a few minutes, it's likely that the spare capacity was affected by substrate availability. If it was done overnight, then it could be substrate availability. Something may have burned out over the period of the incubation, but it can also be a change in mitochondrial production, mitochondrial mass. So your next step might be to do PCR or electron microscopy or something to look at the amount of mitochondria, or run the Seahorse substrate oxidation assay to see if one of the substrates has become more or less dependent for the cell's oxidation. So back in 2011, Erica Pierce started running some Seahorse assays, actually 2009. And one thing that she did is she took a mouse, she infected the mouse, and then she took splenocytes and thymocytes and looked at cells before, during, and after the infection. And what she noted is that the naive cells had very little extracellular acidification, meaning they were pretty quiet, not doing a lot of glycolysis. And when they were activated, she saw much higher glycolysis. This was known at the time, and she was validating the Seahorse technology for use to study cell activation, immune cell activation. Over time, some of the cells persisted as memory cells, and she found that the memory cells look very much like the naive cells. So if you were to just look at basal rates of extracellular acidification, or even basal rates of oxygen consumption, it would appear that there's no difference between the naive cells and the memory cells. But Erica Pierce took this one step further, not only looking at the basal rates, but she ran the cell mito stress test, and she found that those memory cells had this higher spare capacity. And we know that the spare capacity in memory cells is driven by an increase in mitochondrial mass. Carl June's group demonstrated that when they engineered CAR T cells, they showed that those CAR T cells that behave like memory cells always have more mitochondria than other cell types. So using your basal rates and looking at ECAR and OCAR on top of also running a cell mito stress test can give you a whole plethora of information to better understand T cell function and activation. So I'll break here to see if you guys have any questions about those two assays.
Kevin, I don't have specific questions about these two assays, but I have other questions. Can you take a couple?
Sure.
All right, and can you hear me okay?
Yes.
Awesome. So one question that I received is there a kit to address isolated mitochondria?
We do not have a kit for isolated mitochondria, but we have a guru. His name is Manish Bharadwaj. We also have another guru. His name is George Rogers. The two of them have run dozens and dozens and dozens of these assays and have published. We have an app note for this, but no kit. But between Manish, myself, my colleague Jay Dunn, and George Rogers, we could certainly help you get started with isolated mitochondria.
Awesome. And then another question that I received is about the phenotype or the energy map. So bear with me. I'm just finding the question. Sorry. So Ray, regarding metabolic switching and phenotype map, what is the difference between aerobic and energetic at high OCAR?
Aerobic and energetic. Very good question. These are arbitrary. So the reason why we call that one quadrant energetic is because we're trying to highlight that the cells are utilizing both oxidative phosphorylation and glycolysis. You see this in T cells, for example. When they become activated, they ramp up both oxidative phosphorylation and glycolysis. In the early stages of activation, the cells start to take up more glucose. They switch over to glycolysis, but they also have more glucose, which gets converted to pyruvate and funneled into the mitochondria. You wind up seeing increases in both. So that's why we call it energetic. But yes, anytime you increase from base, regardless of what direction you're going, you're making the cells more energetic. So I think it was just maybe the product manager couldn't think of a better word.
Thank you. And then I'm going to take a third question here. Do you recommend using cell adherence products like CellTac or poly-D lysine?
If you need to, if you have a question about your cell line, you can convert our cell line reference database, or you can contact your field application scientist and they will let you know. Most cell lines do not require an adherent. A lot of primaries do. And if you just want to be safe, I would recommend using poly-D-lysine. You can do that every time if you want. It shouldn't have any ill effects on your cells, and oftentimes it benefits.
Okay. I did say 3 questions, but I'm going to take the 4th, Kevin. So does FCCP need to be titrated for each cell type? Example, smooth muscle cells versus endothelial cells and or a specific experimental condition?
The base answer is yes, you should titrate it against all different conditions. There are some things you can do if that becomes problematic. For example, you've just got too many cell types. You could do 2 rounds of FCCP injection sequentially. So you wind up with 2 different doses. That can help shorten your optimization or running of many, many different samples. But we do note that endothelial cells and muscle cells often have different concentrations of FCCP that are effective.
Okay. And what is the lowest cell number that you could put in a plate?
It depends on the cell. Some of the more energetic cells like brown fat and muscle cells, around 10,000, maybe 7,500 to 10,000.
All right. And is there somewhere on the website that could give us a starting point?
There's a cell reference database, which we'll go over soon, where you can get some information about that. We also have assay templates. I think there's 16 of them for the most commonly used cell types. We have neurons, macrophages, HEC293s, C2C12s in there, which tell you what density to use and what you should expect.
Awesome. No more questions for this time period. You can go on. Thanks, Kevin.
Okay, I'm sorry I'm running a little behind, so I'm going to speed up a little bit. I want to go through the glycolytic rate assay. The glycolytic rate assay was designed to look more specifically at glycolysis. The Seahorse instrument measures pH changes, and it doesn't really care where those pH changes are coming from. A majority of the pH change, 80% or more, is coming from glycolysis. The remainder is mainly from the Krebs cycle. So we can model out the Krebs cycle. We can account for carbonic acid, which comes from the Krebs cycle, simply by adding rotenone and antimycin. Remember, that blocks mitochondrial respiration. There's a relationship between the amount of oxygen consumed by the mitochondria and the amount of acid produced by the Krebs cycle. So we can take that information and then subtract it from the overall acid that the instrument's measuring, getting us back to basal glycolysis. So in this assay, we're going to inject rotenone mixed with antimycin and then 2-deoxyglucose to demonstrate how much of this is glycolytic versus non-glycolytic. And we also get compensatory glycolysis. So an example of this from our own labs shows that you can inject a pyruvate transport inhibitor and you'll see an increase in the rates of proton efflux. And these are 2 different cell types, a lung cancer cell and a bronchial airway epithelial cell. And they've got very different kinetics. After we've added the inhibitor, then we add the rotenone, we get a certain level of compensation. And you can see that in the lung cell, the compensation isn't quite as great as in the bronchial airway epithelial cell. That may mean that the lung cell is more dependent upon glycolysis than, say, a bronchial airway epithelial cell. When you run this through a report generator, you can get graphs back like this, not with the lactate data, I'll get to that in a second, but with basal glycolysis without the inhibitor and basal glycolysis with the inhibitor for the 2 different conditions. You'll also get additional data such as compensatory glycolysis, non-glycolytic acidification, and so forth. The reason why the graph is drawn this way is it shows that your basal glycolysis in this data matches very closely with a correlation of about 0.9 with your standard lactate assay. The big difference is that our assays are kinetic. All these assays are occurring in real time. It's a kinetic-based assay. You can run these assays up to 12 hours. You can inject multiple compounds to see how it affects glycolysis. So we expand what you can do if you're interested in studying glucose uptake, glycolysis activation, compared to looking at just a lactate assay. And using these 2 assays I just described, the cell mito stress test and the glycolytic rate assay, will get us to ATP production. So the assay I just showed you is the glycolytic rate assay. And for every 2 protons that are produced, you're making 2 ATP. So the glycolytic rate assay is also an ATP rate assay for glycolysis. Those units are the same. So although it says proton efflux rate, it could just as easily say ATP in picomoles per minute, because for every 2 protons that are made, you're making 2 ATP units. For the oxygen data, remember I said before, you want to calculate that oligomycin response, and then we just need to multiply it by a couple of factors. 2, because oxygen is consumed as a doublet, and 2.75 is the ATP to oxygen ratio for our media. That number does need to be calculated, but we know it's 2.75 for the Seahorse assay media. If you make up your own media, you can calculate it and enter it into the software in order to calculate ATP units coming off.
I don't think so. Actually, I could just go.
So the assay is pretty straightforward. You inject oligomycin, make a measurement, and then inject the rotenone mixed with antimycin and get a couple more measurements. And then this data is going to be sent to Excel. Oh, I got ahead, sorry. Just give me a second. Sorry, my laser pointer got lost. All right, there we go. So this data is going to be sent out to Excel. And the Excel macro that we provide will do all the calculations for you. So you don't have to do these calculations on your own. Once you get the data back, it's going to look something like this. So it looks like an energy map, but now we're looking at mitochondrially produced ATP against glycolytic. On the screen, what you're looking at are estrogen positive and estrogen negative breast cancer cells. So if you have an estrogen receptor expressed, you are likely to be more oxidative than if you don't, in terms of your ATP production. Over here, what we have is called the ATP rate index, where we simply divide the amount of ATP produced oxidatively by the amount produced glycolytically. The index, the higher it is, the more oxidatively your cells are producing ATP, and the lower it is, the more glycolytically. So I understand breaking up, that might just be bandwidth. Am I okay now? Rula?
Yeah, you sound fine, Kevin. Just get closer to the speaker if possible.
Yep. Sorry, I had to make a change to one of the monitors. So my head moved.
No worries.
Okay, so that's your ATP rate index. You can also plot the data as bar graphs, like this one here on the left, which is a screen of many different cell types, which is showing you the rate of ATP produced mitochondrially and the rate of ATP produced glycolytically in red here for all these different cell types. So you can see that this cell type here, the Jurkat cell, doesn't make a lot of ATP per unit cell compared to, say, an HMEC cell. On the right here, what we've done is we've looked at the percentages, like what percentage of ATP is coming from glycolysis versus what percent is coming from mitochondria. That's why the Jurkat cell looks much bigger now. But what it's telling you is that a majority of the ATP is being made glycolytically as opposed to oxidatively in this particular chart. And this was the assay that was used to profile norovirus in a recent publication that we've turned into an application now. So this is available on our website. If you happen to be interested in studying infectious diseases, you can infect cells with norovirus, coronavirus, respiratory syncytial virus, and then look to see how your cells respond in terms of their ATP production. And what they're demonstrating here is that macrophages, raw 267 cells, when they become infected, they increase their ATP production, and there's increases in glycolysis in particular. And if you were to block glycolysis, you revert things back to their uninfected state. So one of the approaches to treating norovirus and coronavirus is to inhibit glycolysis in some way. That's the goal behind that drug W1P22 that I had mentioned earlier. And one of the final assays that I want to talk about before we switch gears and talk about setting up an experiment is the substrate oxidation assay. This is built off of the cell mito stress test, but the difference here is that we are going to add an inhibitor before we add the cell mito stress test. So the inhibitor is going to block one of the substrate pathways, either fatty acid or glutamine or glucose. And right off the bat, we'll see if it's important to the cells. If you see a decrease in respiration due to the inhibitor, that means that that cell required that substrate just for basal ATP production. Then when we hit the cell with the FCCP, you'll see whether or not that particular substrate is of paramount importance because you'll lose that spare capacity. Remember, I said spare capacity is tightly linked to mitochondrial mass and substrate availability. If you don't get the substrate to the mitochondria, you don't get an FCCP response. And here are some examples from our own labs. This kit was just released, so there are a couple of publications out there, but I'm not going to show them. I'm showing the internal data right now. And here what you're looking at are 3 different cell types, and these are charts that came right off of the Seahorse analytics on our website. And on the left is a breast cancer cell, and you can see right here, this is glutamine. If you inhibit glutamine, you lose a response. So these cells are glutamine dependent. Here we've got C2C12 cells that appear to be glutamine and glucose dependent. And over here we've got HepG2 cells, which are glucose and glutamine dependent, and maybe a little bit fatty acid dependent. So this assay will let you know what substrates are getting funneled into the mitochondria and how relevant they are for that particular. All right, that completes the first module. I hope that you guys found it informative and somewhat entertaining. I am going to shift gears and talk about how to set up an experiment. Before I get to that, I'll be happy to answer any more questions you have.
Kevin, we have one question. In mito stress test assay, after adding FCCP, OCAR reaches the plateau. Sometimes the plateau goes up, stays flat, or goes down before the addition of oligomycin. What do those situations mean?
Right. So this data here, you might notice that the rates are going up before they peak. That usually indicates an incorrect dosing of FCCP. That's why we recommend more than one measurement after the FCCP. This was probably just not quite enough FCCP, but over the 3 measurement period, it got up to a peak. This is a better scenario than putting too much FCCP in. In the other case, which I don't have a great example of, but you kind of see it here, it goes up and then it starts to come down. This could also be an incorrect dosing of FCCP, but it can also indicate that you're running out of a substrate. If you're running out of a substrate, this is going to continue to decay until it gets back down to basal or even lower. So those are the reasons why the FCCP responses can go in those 2 different directions.
Thank you, Kevin. Is it possible to apply ATP assays to animal tissues or homogenize animal tissues?
Homogenized, I'm going to say yes, but it hasn't been validated yet. It relies on the ability of the oligomycin and the FCCP to exert their effects. Oligomycin is a large molecule and sometimes doesn't penetrate tissue. So some of the tissue protocols you see won't even use oligomycin. But as long as oligomycin and FCCP can penetrate the tissue, you know the ATP to oxygen ratio, and you use the standard plates, that is the XFe96 V7 plates, the V3 plates for the 96, you should be okay. These assays are not designed to be used with spheroid plates or with the islet plates.
Thank you, Kevin. I'm going to build on that. By not validated, you mean this specific assay has not been performed in our labs. However, Kevin and other application engineers can work with you to help you achieve your goals. So moving on to the next question, what variability should I expect from the assay? How robust are the measurements?
That's actually going to be part of the next section. The measurements are very robust in my experience, providing the tissue culture is done very carefully and your pipetting skills are in good shape and you're very careful with your pipetting. A lot of variability is due to pipetting. Some cell types are a little bit more forgiving than others. I mean, C. elegans is going to have slightly higher variability. In my experience with C. elegans data has CVs of around 15 to 20%. However, in cell lines, the CVs range from 3% to 12%. And the difference is either the cell line or the person doing the pipetting.
Okay. Kevin, I'm getting many questions. So how many more questions can you take at this point?
Let's take 3 more. 3 is my lucky number.
Okay, cool. So when it comes to substrate oxidation test, how do you know the correct dose of each inhibitor?
The FCCP needs to be titrated. The other inhibitors work at the concentrations we recommend for most cell types. I would not change the concentrations of the Baptist, Etamoxor, or UK5099 without consulting with us first. Those concentrations have been studied in a lot of detail over the last 6 years, so we're pretty confident in those. The only one that needs to be titrated is the FCCP.
Okay, thank you. So question number 2 out of the lucky 3, what is the range of acceptable ECAR values? Are cells with low ECAR unhealthy? When to use ECAR or PER for glycolysis data presentation?
The ECAR range is 5 millipH per minute to much higher than the cells could ever achieve. If you have 5 millipH or less, it doesn't mean the cells are unhealthy. They just may not be very glycolytic at all. I think some cell types are extremely oxidative. Some pancreatic cells, for example, I think beta cells are very oxidative. So most of the glucose, if not all of it, gets converted to acetyl-CoA. So that's not a bad thing. What you want to do is check both OCAR and ECAR. If they're both low, that could indicate that the cells are unhealthy or just not very energetic. The choice of when to use OCAR versus PER is more or less up to you. However, PER is required for the glycolytic rate assay and the ATP rate assay. I also believe that the PER data, actually I don't believe this is known, the PER data is more quantitative than the ECAR data. ECAR data is on a logarithmic scale, so it's on a whole other ball of wax, and it's also sensitive to pH. So if you're using your own media and you're not careful about pHing your media, then your ECAR data could change from run to run. However, the PER data is corrected for by the buffer capacity. So as long as you know the buffer capacity and your media's pH is checked, your PER data will be very robust and quantifiable using absolute measurements.
Thank you, Kevin. So final question for this time period. It's a 3-part question, so I'm going to speak very slow. All right, I'm going to get started. Looks like Cyclant is suggested for DNA quantification after these assays for normalization. Is there an exact protocol for this following Seahorse assay? For instance, A, freeze plate following Seahorse and removal of assay medium. B, treat cells with RNAs before Cyclant. C, done in adherent cells or suspension cells. And what is your preferred normalization method?
Excellent question. I said that there were 4 questions we get quite a bit. One is your software compatible? The other is, where did the Seahorse instrument get its name? The next one would be how many cells to use, and then the next one is how to normalize. We have many protocols for normalizing that utilize host or DAPI and then a cell counter. In fact, we currently recommend using a biotech citation 1 or 5, labeling your cells with DAPI either during or immediately after the assay, and then letting the plate reader do the lion's share, or the plate imager do the lion's share of the work. It can calculate, it can collect the images and then count the number of cells. And I actually have an example of that at the end of the next module. As far as Cyquant is concerned, we don't have an official protocol. I have one that was given to me by a friend, and I can see if he's okay with distributing it. As far as I know, he simply followed the manufacturer's recommended instructions.
All right. Thanks, Kevin. No more questions for now.
All righty. So would now be a good time to take a 5-minute break, do you think, Rula?
Absolutely. I need it, and I'm sure the audience needs it as well. So let's circle back at 11:15, everybody.
Okay. And just so you all know, I'm going to start by showing some old photographs. So I'm doing this on purpose in case you're a little slow to get back. You might need to take care of something, drop off the mail, if you've got some kids. So don't feel a need to rush back. The photos are fun. They'll take about 5 minutes to go through. So I will start with those at 11:15.
Kevin, I thought you were going to talk about infectious diseases. Just kidding.
No, that's on May 27th.
All right.
I can address that.
See you all at 11:15. Thanks, Kevin. Kevin, I am back.
Excellent. Can you hear me?
I can. We still have one more minute, though.
Right.
I'll alert you when we can get started. Nice job so far. Everything is going nice and smooth.
Good. I hope it's helpful.
That's the goal. We're doing the best we can, given the circumstances. All right, it is 11:15 and we're going to go ahead and get started. Let's pick up.
So hopefully you can see my screen, and what you're looking at is an XF24 or XFE24 during assembly. So this is what the inside of the instrument sort of looks like. These parts here will eventually be threaded behind here. This part is, actually this is an, this might be an E96. So this part here is called the probe head. It houses the probes. The probes are here. Then, yeah, it does look like a 96. And you see the light pipes right here. On the bottom of the light pipes is a lens, and threading off of the light pipes are fiber optics that are then ensheathed in a black, well, I forget what they call this stuff. We use it for our wires, electrical wires. So that is encased in that to protect it from light, and then that feeds through a comb that then inserts into a board right here. It's not shown, but behind here is something called an electrical optical board. That board manages all the light. It transmits light using an LED, and then it reads back the emitted light from the sample, from the fluorophore, and then does the digital analog to digital conversion, and then sends that off to your controller so you get your rate data. This one, since it's under assembly, doesn't even have the stage on it yet. There'd be a stage here for your cell culture plate as the build continues, and of course they would put a bezel on it at your cover to keep it nice and light tight. Here's another picture of the fiber optics, a little bit closer up. This is the top. You see they're on a spring. So they go through a spring into the probe head that allows them to easily go up and down during the assay. This is a picture of an XFE96 cartridge. It'll eventually be colored green, but this one, it's clear, and you can see the spots on the bottom here, the big white spots there. And this is a picture of them assembling the cartridges. We actually have a device called a spotter, I think they call it the printer, where you place the cartridges, and then this little arm goes around and squirts a mixture of the fluorophores and the polymer onto each one of the ends of the sleeve. So here's another picture of that. And I think they said it takes about 6 hours to make a flux pack. So that's 18 cartridges, all with their spots. And I think we've got, actually, I don't know how many of these we had. When we were Seahorse, we had 3 of these. I don't know if we have more now. But this is the device that actually makes the cartridges for us. And the manufacturer is done in Chicopee, Massachusetts. And this is what happens when people get together to discuss Seahorse data. This was, and I hope we can get back to this sometime soon. This was our first user group meeting, which took place in New Hampshire. And our keynote speaker was David Nichols that day. This is John Lemasters. I can't quite tell, but this might be David Farrick, our Chief Scientific Officer. And when we do these workshops, we do these regionally now. Back then, we were too small to do them regionally. We didn't have enough users. So we held them in various places in New Hampshire and New England. All right, so let's get back to it. All right, so again, if you're just joining or you weren't sure how to ask questions because everybody's been muted, chat your questions, text your questions to Rula. And Rula, can you see my presentation screen or my notes screen?
I can see your non-presentation mode PowerPoint.
Oh, let's see. And now?
I can still see the same thing.
Oh, so you're seeing my notes.
I'm seeing your PowerPoint. I'm not seeing your notes. I'm seeing your PowerPoint, no notes, and it's in a non-presentation mode. I can see the slides on the left.
Oh, okay.
And the slide in the middle.
How about now?
Yep, I can see the full slide. Perfect.
Great. Okay. Sorry about that. All right. So let's talk about setting up an assay. All right, so there are a few basic steps that you need to accomplish in order to set up an assay. You're going to need cells or some sort of biological material. You'll note on our cell reference database when we get to that section that we've got over 1,000 different cell types that have been utilized, and well, maybe not quite 1,000, but at least 600. And this includes organisms like zebrafish embryos, C. elegans, bacteria, among other things. The material that you're using needs to find its way into a Seahorse tissue culture plate. These plates are specially designed with this little bevel up here to accommodate the fiber optics. Your material should be immobilized on the bottom of the plates, and when you transfer them into the plate, you're culturing them as you would normally. So if you do any type of passaging of your cells, it's all you're really doing here is a secondary culturing, passaging of your cells, putting them in the Seahorse instrument, Seahorse tissue culture plate, and allowing them to grow until you're ready to use them. Most people seed their cells in the Seahorse plate and grow overnight. That's going to be in your standard tissue culture incubators with your standard media. No changes need to be made here. You just need to get the cells into the Seahorse plates. When you're ready to run the assay, whether it's hours after seeding, days after seeding, the longest I've ever heard was about 4 weeks when someone was doing iPSC-derived cardiomyocytes. When you're ready to run the cells, there's a media exchange step. The media exchange step is to remove the bicarbonate. If you purchase media from us, we have RPMI and DMEM, which is weakly buffered and does not contain any bicarbonate. This is important because bicarbonate will cause your media to alkalinize, and we do not want the pH to change during the assay because it'll be more difficult to measure glycolysis. Not only that, but it may not be healthy for your cells. This does not mean you can't run assays at an alkaline or an acidic pH. You can, you just don't want the pH to change during the assay, and you want to be mindful of what bicarbonate can do during the assay. So we're going to swap the media and put it into a bicarb-free, low-buffered media. The volumes here after the media exchange should be 500 microliters per well in an XFe24 and 180 microliters per well in the HS Mini or the XFP. This plate will go and sit in a non-CO2, like a bacterial incubator, without CO2 at 37 degrees for a minimum of 45 minutes. That period of incubation without the CO2 is to also make sure, in addition to not having bicarbonate, we don't have any CO2. CO2 is a gas that can be in the plastic, so we put the plates in a non-CO2 incubator to allow the CO2 to outgas. CO2 will cause acidification, again, making it difficult to get good reads on your glycolysis data. It takes about 45 minutes for the CO2 to dissipate. So that's what we recommend at this step. And this is all on our website, and I'll show you where to find it. In fact, most of these slides have come from our website. Over here is our secondary. We need to prep the cartridge. In order to prep the cartridge, the way this was designed is the spots are dry and they are impenetrable to oxygen or to proton. So they won't work when they're dry. They're also shipped in a foil packet, so they're protected from light. You'll open up the foil packet, you'll find out that you have multiple parts. You'll have a utility plate and you'll also have the cartridge, and depending upon what instrument you're using, you may have some other bits and bobs too. But we're going to focus right now on the utility plate and the cartridge. The utility plate is basically a cell culture plate, but it's not cell culture treated. It's designed simply to carry the cartridge around and also to hydrate the cartridge. So there will be a step where we're going to hydrate the cartridge overnight. So we're going to put 500 microliters of calibrant or 180 microliters of water, if we're using the XF96, into the utility plate, and then let the cartridge go overnight in the non-CO2 incubator. Minimum time is 4 hours. Maximum we recommend is 72 hours, and the maximum is limited just because we're concerned about evaporation. If your material, if your fluid evaporates, you're going to get air bubbles, and that's going to wreck your oxygen data. We recommend overnight, but the other parameters are fine if you need to do them, 4 hours to 72 hours. Right before you run the assay, you are going to load whatever drugs you want to use into the drug ports. Because there's no perfusion in the instrument, because you're injecting fluid into fluid, you've got to be mindful of the fact that whatever you inject will get diluted. So make up concentrated stocks. Our protocols for all of our kits, we have you make up a 10x stock. So for example, if you found that 1 micromolar FCCP was optimal for your cells, you would make up a 10 micromolar stock and then load 20 to 30 microliters of that into the injector ports. You'll then program the instrument and then start the assay. When you start the assay, the instrument will prompt you to load the cartridge in the calibrant. So make sure cartridge and calibrant goes in first. Many of these steps can be automated. If you've got a liquid handling system, Agilent manufactures something called the Bravo, which is very nice, doing a lot of these handling steps for you. And this makes very accurate pipetting and helps to reduce your error with these tools. They work very, very nicely. This is sort of a walkthrough, again, of seeding your cells, washing them. You can take photographs of them. I always recommend looking at your cells before you start the assay. In this picture, it shows the cell plate going into a citation 1 from biotech for photographing. If you don't have access to something like this, I do recommend at least looking at the cells in a microscope to make sure they're there, they haven't detached during the rinsing, or they haven't died. Make up your compounds, add the compounds to the ports, and then start the assay by loading the cartridge on top of the utility plate, and make sure the utility plate is in calibrant. Now, you may have picked up on the fact that I used the word water for the XFe96 and the XFP as part of your hydration, and I said calibrant for the XF24. They are different, and you'll see why in the next 2 slides. For the XFe96 and for the XFP, we recommend hydrating in water. The reason is that when you put the 200 microliters of water in each of the wells and then put the cartridge on top, the cartridges sit pretty low in the wells. So those spots are awful close to the bottom of the plate, and a pocket of air can form as a result. If that happens, then you don't get proper hydration. So what we recommend is that you hydrate your cells in water, put some calibrant in the incubators next to them, do the overnight hydration, and then the next day, dump the water, and you can simply flick it in the sink if you want, and then replace it with calibrant right before you start the assay, right before you load the plate with your drugs. So this will ensure that you don't get any air bubbles lodged in the bottom, and will help improve your calibration and your data quality. So these are the steps for the XFe96 and for the XFP, or the HS Mini. Now the HS Mini, and it's not shown here, but they have one other little part to them. Surrounding each of the wells is a moat, and we recommend putting 400 microliters of tissue-grade water into those moats. And this helps prevent evaporation. This is the only plate that has a moat. None of the other plates have a moat. So if you don't use a 24 or a 96, then you can just ignore what I just said. But if you are using an XFP, it's important to put water in that moat to prevent evaporation. Now for the XF24, it comes with a special plate that we call the Hydro Booster, which is a riser. Remember I said with the 96 that the cartridge sits awful low. Well, in the 24, they designed this extra piece of plastic called a Hydro Booster to raise the probes up so they don't sit at the bottom. This prevents air bubbles from forming. As a result, with the XFe24, you can hydrate in calibrant as opposed to water. So you just put the cells in calibrant, and then when you're ready to start the experiment, you've got to remove that Hydro Booster. So pull the cartridge out, take the hydro booster off, put the cartridge back in the plate, and make up your drugs and load them into the drug injector ports, and you're good to go. For seeding, this is one of the more important steps of the whole assay. The two places where most of the variability occur are here and in the making of the or loading the drug injector ports with the multi-channel or single-channel pipetter. So we do have some recommendations to help improve the cell adherence. So this is not a mistake in this picture. You see how the pipette tip is angled here? What really helps with the seeding is to make sure you accurately count the cells and hopefully make sure it's a viable count. Some people work with frozen material and the viability can be all over the place when you thaw those cells out. When you've got greater than 90% viability, you're in really good shape here. So hope that you have, or make sure you have viable cells, and then do thorough mixing in between each of your pipetting. I can't emphasize this enough. The cells will settle, or they will distribute unevenly in your trough or even in your flask. So I always recommend doing a lot of tapping, mixing, to triturating, just to make, and that's pipetting up and down, to make sure that your slurry is nice and well mixed in the trough before you do the dispensing. Now I said that this is not an accident. Once you have the cells in your pipette tips, we recommend angling the tips so they touch this little lip here and then eject the cells into that lip. What happens is the cells will hit that and they'll ricochet and they drop right down into the center. So this really helps getting an even distribution of cells from well to well, and that will help reduce your variability. When you seed the cells, we recommend seeding in a low volume. So we're talking about the XFe96 and the HS Mini here. So the seeding volume is 80 microliters. Remember, when you rinse your cells, you're going to take the 80 microliters away and then replace it with 180 microliters. So your final running volume is 180. But we seed in a low volume because we don't want the cells to grow up here. We want them to grow down here. So you seed your cells. Then we recommend letting the plate rest for 1 hour in your tissue culture incubators. In your tissue culture hoods, not the incubators. The reason is we want them to rest at room temperature to even out the temperature across the entire plate. Because when you put the plate into the incubators, it's going to start to warm up. And if there's differential heating in the plate, for example, you go to seed your cells, the edge wells cool off faster than the center wells, because you're keeping all of your media as warm as possible during this whole procedure. So it may take a while to seed the cells, and the edges are going to cool faster than the center. That leads to differential evaporation if you suddenly put it into an incubator. I mean, you can try this experiment at home if you want. You basically just put water in a dish or a couple of dishes, let them sit at different temperatures, then put them into your oven, and you'll see that they'll evaporate at different rates. So it's the same idea here. The evaporation can lead to a lot of well-to-well variability. So letting your plate rest for 1 hour in your hood will help prevent that from happening. Then you put the cells into your tissue culture incubators and you culture them until you're ready. For the XF24, the procedure is the same, but the volumes are different. We recommend seeding the cells in 100 microliters of fluid, let them rest for an hour. It says 1 to 6 hours. That's because you want to make sure the cells are adhered, and then you bring the volume up by adding another 150 microliters. You could do the same thing with the XF96. In other words, you could wait just a couple of hours for the cells to adhere, then bring the volume up to 180 if you want. For the XF24, we found that this step really helps to reduce variability. We call it the two-step seeding process. After you've brought the volume up, so now you've got 250 microliters total, you can place your cells into the incubators and let them grow until you're ready to use them. Now, if you're going to let them grow for more than 24 hours, I would highly recommend doing a media exchange and then carrying the cells in the maximal volume, which would be 500 microliters or 1 mil, whichever you're more comfortable with. And that again is also to prevent evaporation. Same goes for the XF96. If you're going to culture your cells for more than a day, make sure they're in a high volume, between 200 and 300 microliters. Otherwise, you're going to get a lot of variability due to edge effect and uneven evaporation. And I often recommend doing half rinses. So my background was in developmental neurobiology, and I cultured lots of neurons. You never completely rinse a neuron in culture, because neurons produce their own trophic factors that support their own growth. If you do a complete rinse, then you wash that away and your neurons will die. So what we did were half rinses. I would culture my cells in 24 and 12 well plates, and I would remove half the media per well and then add half fresh back. And that's the procedure I recommend if you're going to carry these cells for more than 1 day. So that is seeding, and I'll break here to take more questions.
Hey, Kevin, I have 3 questions. So your lucky number. Can you hear me okay?
Yes.
Okay. So first question, I wonder if we could reuse the calibrant solution.
We actually have never tested that. The answer is likely yes, but I don't think you would need to, because we ship a lot of the calibrant with the flux packs with the cartridges. So when you buy cartridges, you get calibrant and more than you would need for the 18 or 6 cartridges that you purchased.
Okay. So someone heard that using DH2O as hydration solution instead of calibrant. Can you comment on that?
Yeah, we used to recommend hydrated calibrant, but what was happening is that some people would calibrate longer than others for various reasons, and they were seeing a lot of evaporation in the calibrant during the hydration procedure. And what occurred is they got pockets of air, we call them micro bubbles, that would form on the spots that do the fluorescent reads. And if you've got a little air bubble there, it throws off the oxygen calculations and you wind up getting negative OCAR. We found that simply replacing the calibrant with water, allowing for evaporation to occur, but then dumping the water, replacing it with calibrant, takes care of any bubbles that form and helps keep the sensor nice and well hydrated, and helps keep volume even across the plate during the calibration step, which helps improve the calibration. So there were lots of reasons for that change. So some of you may have used an instrument years ago prior to 2016, and we recommended hydrating only in calibrant. Now we recommend doing water, then switching to calibrant.
Thank you. And then can you please have Kevin, can you please address issues of mycoplasma contamination and or excess cell line passage issues, for example, extraordinary artifact results?
So with mycoplasma, the funny thing is, in the 12 years I've worked with Seahorse, I've probably seen mycoplasma more than I realize, but I haven't seen that much contamination. I've seen some bacterial contamination, which gets into the media in some cases, not in the tissue culture incubators. And whenever you run these assays, we always have 4 background wells. These are negative control wells that don't have any cells in them, just media. So in those cases where the media was contaminated, you actually see rates of respiration in those background wells, which lets us know that the media was contaminated. In those cases, your Seahorse data winds up being highly variable. So you get a lot of variability. So that usually is a tip-off that something might be wrong. High variability and rates in the background wells. For mycoplasma, to my knowledge, I've only seen it once. And what it did is it completely changed the phenotype of the cells that the lab were running. They were running A549 cells in the cell mida stress test and getting pretty solid data. Then one day they go and run an assay and the spare capacity just didn't exist anymore. And the variability was much higher than they expected. So they were running assays and getting about 8 to 10% variability. All of a sudden they're getting 15 to 20% variability and no response to FCCP. We then go through a number of things with the lab to try to figure out what's going on. Did they count the cells right? What was the passage number? How does this compare to previous experiments? What was the ECAR data like? And everything in this case checked out. Everything was done properly. So one of the last things we recommend is to check for mycoplasma. They did. They found out it was mycoplasma positive. So they cleaned out their incubators, started over again. Data were much better. As far as passage number, I've seen lots of effects on passage number. Earlier in the presentation, I showed you the C2, C12 data. or a cartoon of the C2C12 data. Oftentimes when cells hit higher passage numbers, they also start to lose that spare capacity from the cell might stress test. So sometimes when I work with bioprocessing people or people that are interested in how passage number affects cells, I recommend the cell might stress test and a focus on that spare capacity.
All right, thank you, Kevin. I have a fourth question, and I'm happy to take that, Kevin. So someone is writing a review paper that has a large section about choosing the right Seahorse assay, pros and cons to Seahorse technology, et cetera. Do you think Kevin or someone at Agilent would be able to go over a few questions with me before we submit? The answer is yes. And in addition to that, later this month, we have an event to help you with grant writing. We have experts on both ends, from the NIH end and from the Seahorse user end. So if you're interested, feel free to send me an email and I can send you the link once it's created. All right, Kevin, so you can move on.
Okay, so I mentioned you have to exchange the media, and the media should be a bicarb-free media. Agilent manufactures DMEM pH 7.4 and RPMI 7.4. The media is buffered with heaps, and we also make substrates that can go with it. Our media is substrate free, and that's on purpose because you're all interested in metabolism and mitochondrial function and glycolysis. So you may be interested in how substrates affect those parameters. So our media doesn't contain any substrates. You can add to it what you want. We do recommend for most of our assays to add 10 millimolar glucose, 1 millimolar pyruvate, and 2 millimolar glutamine. That is standard for most Seahorse assays. You change that if you have an interest in looking at glucose deprivation, or if you have an interest in fatty acid oxidation, you might add some palmitate to the media. Some people that are interested in diabetes and obesity will put higher or lower concentrations of glucose to simulate a diabetic condition. But for most assays, 10 millimolar glucose, 1 millimolar pyruvate, and 2 millimolar glutamine is what is recommended. And those numbers are saturating. That's why we came up with them. It's more than the cell would need. That allows the cell to adjust to anything you throw at it. When you add the FCCP, hopefully you won't run out of anything. But if you happen to have a cell that is highly dependent upon glutamine and it races through the glutamine before you start the assay, then your FCCP response will be low. So we make sure that that doesn't happen by including high concentrations of these. Now, we do recommend not using serum. That's a recommendation. The reason is that serum tends to foam or froth. It makes air bubbles. I personally work with people who use serum. I coach them about it. Just need to be mindful of this. I recommend if you do want to use serum, drop it down to 1% and just be careful with the pipetting. When the assay is complete, look in the wells to see if there are any air bubbles that formed or any foaming. That would be a giveaway that you might get some bad data in that well because of pockets of air forming. So the method for rinsing, we suggest you look at the cells before you rinse them to make sure that they're happy and healthy. And then we remove most of the media and then add the new media to the well, and then let it sit for 60 minutes before we run, well, 45 to 60 minutes before we start the assay. It's important, no matter what you do, to be consistent with all of these steps. Consistency is key. I've seen people that were consistently bad and they still got good data because they kept making the same mistake over and over again. So consistency is really important. So whatever you do, try to be as consistent as possible. And we do recommend using the plate imager to take the photographs. They can photograph bright field as well as fluorescence, which is going to be great for doing your normalization later on. Liquid handling really helps to reduce variability because of the accuracy of the pipetting. So I want to talk now about one of the most critical steps of running the assays, especially for the users of the XFe96. The XFe96 is a great high throughput instrument, but it's got a lot of drug ports loaded. So this picture here shows you the top surface of a XFe96 plate. This is the layout for the ports, the A port's in the top left, the B port top right, C bottom left, D bottom right. And then this piece here is called an injection bay. Only the XF96s come with the injection guide. Over here is the recommended starting volume and then the volumes that you should load into the ports. Excuse me. The guide is designed to help you load the ports because these holes here will only be over one of the 4 ports and they will fit with your 8 or 12 channel multi-channel pipetter. The proper technique for using this is to drop the pipette tips into the loading guide and then bring your hand straight up. So this picture shows them going in at an angle. We don't recommend actually dispensing at an angle, just to get the tips in. You then bring this straight up, and notice how the tips are not the fine tips. These are just your regular run-of-the-mill P200 tips. If you use tips that are too fine, you run the risk of dropping the tips too far into the wells, and you could actually dispense the fluid right through the port. So you bring them in at an angle, bring your hand straight up, and let gravity pull the tips in, and then dispense the material. And ideally, what you are trying to do is form a seal of fluid in the ports, like this, or like this. If you use a tip that's too thin, it could go all the way to the bottom, and you could accidentally inject your fluid right through the ports. There is no gasket here. The ports have a small hole at the bottom that's 400 microns in diameter, and the drug is held in place by capillary action or surface tension. So if you go too far in, if you're too forceful, you could eject the contents right through the port. So I always recommend trying to use the smallest possible pipetter. A P50 is ideal for this. If you don't have a P50, try a P100. If you don't have a P100, P200 is okay. P300s don't work very well, and anything above that's not going to work very well. Probably don't use a P300. Most people have a P200. But make sure tips go into the injection guide and they don't get stuck. If they're getting stuck, that's a bad sign. If they're getting stuck, you could always load without the injector loading guide, but it's a bit more cumbersome. In this case, you do angle the tip so it rests in the top third of the port, and then you allow the fluid to cling to the side and then grow as you dispense it. You also need a minimal volume. If you seed less than 200 or inject less than 200 microliters, the ball of fluid may not form a seal. If it doesn't form a seal, then when the injection occurs, the air just passes right through it and you don't get an injection. Lastly, because this is a very sensitive part and requires surface tension, load new instrument. Don't load your drug injector ports in another building and then walk everything over. By the time you get there, you will likely have broken the surface tension simply by opening doors, shaking hands with somebody, if we're allowed to still do that, or accidentally bumping into something. You could break the surface tension and then the fluid would drop through and you will get a failed injection and all kinds of messy data after the injection occurs. So these are steps that should be taken with care with the XFe96. And with the XFP and the HS Mini, you don't have a loading guide. You're only going to load this way, but you don't have that many ports to load, so it's a little bit easier to determine if things are going smoothly. Now, it's hard to tell if you've got a seal here. What you can do, and I wish I could show you this, is if you look straight down at the ports, the light from the room will reflect off the fluid sitting in the port, and that port will glow. It'll be a little bit brighter than ports that aren't loaded or aren't properly loaded. So your quality control check is to look at the brightness of the ports. If they're dark, they're not loaded. If they're bright, they're properly loaded. Now with the XF24, and I don't even, did I skip it? With the XF24, yeah, we don't even put it in the guide. So with the XF24, you're basically loading using this method here. But the ports are much bigger, much easier to see, and you'll have an easier time of loading. There's no loading guide. You can use a 8-channel, or I don't know, you couldn't use a 12-channel, too many channels. Well, you could, but you wouldn't use all the channels. When you use an XF24, you would use an 8 or a 12-channel pipetter, but only every other pipette tip will be loaded, and that will allow you to load all the ports of the same type simultaneously. Now, a couple of things to note about the loading. You got to load the entire plate for each port that you intend to inject. So if you're running the ATP rate assay, that's a 2-injection assay. You're using ports A and B. every single A and every single B must be loaded, even if you're not seating cells in the entire plate. The reason is you get one puff of air for all 24, 96, or 8A ports. The air is distributed evenly across all of them and then is pushed on through. If you leave a port empty, the air escapes through the path of least resistance, your air pressure drops, and you'll get a failed injection. So make sure you're always loading all of the ports that you intend to use. That is very important. So to review, this is a very sensitive part of running the assays. Carefully load the ports, use a loading guide if you can. If it's troublesome because the tips are getting stuck, you don't have to use it. There are no loading guides for the XFP, the HS Mini, or the XF24, and use the room light to let you know if you've properly loaded. Now with the XF24, because the ports are bigger, you can hold it up and look under and see very clearly if all the ports are loaded. Much harder to do with the XF96. And then lastly, remember to load all of the ports. The injection volumes were listed on the previous slide for the XFP and the XF96, and you'll notice they go from 20 to 28. Remember, there's no perfusion, so when you inject, you've changed the volume in the well, you increase the volume. For the XF24, you start by injecting, I think, 50 microliters, and it goes up from there. I'm sorry I don't have the chart for that. I forgot to put it in the presentation, but it's online, and we'll go and find it in a few minutes. And that is the basic setup. What I'm going to do next is open up the software and show you how to program the software. Are there any questions at this point?
Kevin, we have 2 questions. I'm going to go ahead and get started. The first question is, can I use a multi-channel pipetter, and does it matter which pipette tips I use?
Yes, you can use a multi-channel pipetter, and yes, the pipette tips matter. I prefer using the standard sort of thicker thicker pipette tips. I have some catalog numbers I can recommend, but it's not on the top of my head. Nothing fancy about them. I wouldn't use the PCR tips, you know, the real fine ones. Just use your regular tips. And if you can, use a P50 or a P100 pipette.
Okay. And a long question, but I'm going to read it slowly. It's actually 2 questions. So first question is, in regards to supplementing the XF base medium, should you try to match the supplement concentration to that found in the original culture medium for the cells? For example, I work with primary cells that are regularly cultured in medium that does not contain pyruvate. Should I leave pyruvate out of the XF medium?
That's a very good question. I would need to talk to you about your assay. Generally speaking, there's 2 schools of thoughts. First, the assay itself only lasts about an hour and a half. So one school of thought is you put the cells in the Seahorse media, they're only in that media for an hour and a half. Whatever phenotype that they have generated in their culture conditions will remain for the duration of the hour and a half assay. That's school of thought number 1. School of thought number 2 basically criticizes school of thought number 1 and says that that guy is crazy. That once you switch your cells into this other media, there can be immediate physiological changes. You can test this empirically, obviously running a Seahorse assay with and without the pyruvate. But school of thought 2 would recommend trying to keep the media conditions as closely matched as possible to your original culture conditions. So yes, you can run the assay without the pyruvate. And if you think it's, and this is why I said I need to talk to you about your assay, if pyruvate has significant effects on your cells, then I would recommend taking it out and running the assay without the pyruvate. People have done that. It's not going to kill your cells, especially if they're already used to it. The other thing is that if they've never seen pyruvate before, they may just ignore it once it's there, or they might think that this is great. You know, I mean, like the first time my kids got to eat a piece of candy, it was, or cake, it was like mayhem. And your cells might respond the same way. So you can test it empirically, or you could try to play it safe and run without the pyruvate.
Thank you, Kevin. I made a note of the person who asked this question, so we will follow up with you separately just in case You have Kevin's email address. I did chat it in earlier. You also have my address as a backup, and I'll keep reminding you of the email addresses as we proceed with this training to set up a follow-up meeting with you. Kevin, the second question I have is, what do you recommend for immune cells or other cells that are not adherent? Should you initially plate in the wash media?
We have a separate protocol for immune cells. What you would do is you would isolate the cells, quantify them, and then spin them down and resuspend them in the Seahorse assay media or your own media without the bicarb at the density and concentration you want. So typically you want to seed about 200,000 cells per 100 microliters. So that's what you would resuspend them in. Then you would dispense 100 microliters of your cell slurry into each well of the Seahorse tissue culture plate. Then you would take that plate over to a swinging bucket centrifuge, balance it against a control plate, and spin it for 5 minutes at 200 Gs. The cells will stick to the bottom of the plate. Now, a lot of people will coat the plates first with Celtac or poly-D-lysine. That's our protocol, is to coat with either of those two before you do the dispensing and the spinning. But I know some people that just spin them without the adherent molecule. You'll have to test whether it works with or without. So that's the protocol. We don't use any braking during the spin, and that's because braking occurs by a little stop and go, which can cause the cells to slide, and we don't want the cells to slide. So we don't use any braking, which means a 5-minute spin at 200 Gs without braking is actually going to take about 20 minutes. When the spin is done, we spun in 100 microliters, but remember the volume for this plate is 180. This is a 96 well plate. So we then will top it off by adding 80 microliters again of our assay media, and then put it in the tissue culture incubator for 30 to 45 minutes just to let it warm up. You want the plate to be warm when it goes into the instrument, the assay. The instrument itself is also warm. Any temperature changes that occur during the assay can lead to variability. Now, the same protocol applies for the XFP, same volumes, and we have a special carrier that fits with your swinging bucket centrifuge. So we have an XFP, and you don't have those carriers, let us know. We can we sell them, but they come with the instrument. So if you're the instrument owner, you probably have them somewhere, and I'll let you know what they look like. And if you're running with the XF24 instrument, the volumes are different. I'd have to check, but I think we increased the seeding volume to 150, but it might actually still be 100. I'd have to double check. But it's in our protocols, which I will distribute to everybody at the end.
Kevin, how are we doing on time? I have 2 more questions. Do you want me to do it now or shelf it for the next time block?
I think we're doing really good on time because the next 2 parts will go pretty quickly. We're going to talk about programming the instrument, then using the software, and that'll probably take about 45 minutes.
Okay, perfect, perfect. And then you have a follow-up meeting next week on data analysis and interpretation, correct?
Yeah, I meant to show you that link.
I shared it. I just wanted to confirm that it's next week. I shared it with the audience already.
So funny thing, I don't know why they did this, but the people who built the house put the doorbell in the office. So that was the doorbell that just rang. Our refrigerator is getting fixed today.
All right, so I'm glad your refrigerator is getting fixed. I'm gonna ask the 2 other questions since we still have time. So when injecting multiple compounds, following the injection of each drug sequentially, is there a concern of the previous drug lingering around in the well with cells? Is there a mechanism by which the previous drug's impact on the cell is neutralized after the readings are performed and before the addition of the next drug?
Excellent question. And no, there's no perfusion, so the drugs will accumulate in the wells. In the kits that we have, we know that the drugs don't have any cumulative effects. I can't say that that's true for something you might be screening or something you're working with independently. There is a chance that one drug could react with another drug. There was a publication by a fellow named Paul Brooks a few years ago where he did a tox screen using a Seahorse instrument. And he actually, I think, triple or quadruple loaded each well. So that means he used the injector ports to inject the toxins, and he just kept adding them. and he did a mathematical model to demonstrate whether or not the drugs had any cumulative effect. I can pull that paper and send it to you, but there was some math that could indicate whether or not two drugs are having some sort of synergistic effect.
Okay, perfect. And I made a note with a follow-up action item to send that paper over. And again, for the person who asked this question, you have our email addresses. Feel free to follow up on your end as well, just in case. All right, last but not least for this session, for the XF96, if I'm only using 2 drugs to fill port A and B, should I fill port C and D with just water or media then? This is to avoid the air bubble issue.
Right. In that case, you're only using ports A and B. You do not need to load the C's and the D's. You only need to load the ports that you're going to inject from.
Wonderful. And by this, I don't have any additional questions, and we are doing good in addressing all questions so far. If I missed anything, please alert me, everyone. Thank you. Kevin, it's back to you.
So I was playing around with my PowerPoint, so hopefully you can see the normalization document on the screen.
Yes, I can. Sorry, I was muted, Kevin. I can.
So I wanted to finish this section with normalization. We have a normalization protocol. My background, like I said, was developmental neurobiology, and I did a lot of immunohistochemistry. In fact, I raised antibodies in goats, sheep, and rabbits. I actually had animals in the vet school at University of Connecticut. Yeah, if you ever call me or email me, I've got some funny stories to tell about that. But I did a lot of imaging, and I personally like using cells to normalize against. You can normalize against protein, and this is external normalization. When you get your data back, I think it's important to normalize your data by counting something external. The amount of protein in the well, the number of cells in the well, the overall fluorescence based off of calcine in the well. All of these techniques have been used to do normalization. My favorite by far is to use Hoaxed or DAPI because it's easy, it's quick, it's clear. So the protocol we have will have you actually load Hoaxed into one of the injector ports, the last port that you're using, and it can be mixed with drugs like rotenone and antimycin. You inject the rotenone, antimycin, and the Hoax, the cells take up the Hoax, and then their nuclei fluoresce, like in this picture right here. The laser pointer's gone, as in the picture up here. So these are all nuclear labeled cells, and then software can easily find them and count them for you. In this particular solution, we actually have linked the Biotech Citation 1 and 5 to the Seahorse software. So Seahorse can control the imaging and acquire the data and then embed it into your Seahorse Wave software. So you don't have to bother with counting the cells, entering the values in, and then doing the math. It can all be automated if you've got a Citation 1 or 5 along with the appropriate controller and software. If you don't, you can still count the cells and enter in the data. This is just something that we created to make it easier and quicker for people, especially folks that do this quite a bit. And these imagers aren't limited to just counting Seahorse data. You can do volume, not so much volumetric, but you can do area measurements, migration assays, 3-4 channel imaging. So if you do any immunohistochemistry, it's a great tool. They can also be used to do ELISAs, among other things. But this is the protocol we have, and we have other modules on normalization. If you have an interest in more on normalization, let us know, and we can share those with you, maybe even do a webinar just on normalization. I think we have 1 or 2 already. All right, so we're going to move on to the software. And I want to remind everybody to chat your questions to Rula because I will no longer be using PowerPoint, so I won't be able to show this slide again. So if you do have questions, chat them over to Rula, and then we will answer them. All right. So is now a good time to take a 5-minute break?
Sure. We're doing good with time. Absolutely. Let's circle back at 12:10.
All right. Sounds good.
Thank you. Hi, Kevin. Nope, that's not you. You're muted.
Hello.
Hey.
So what's on the screen right now?
I see really cool little cartoons and pictures.
Cool, excellent.
Cell biology game.
This is the game I was telling the group about 2 weeks ago. So I'm a big board game geek. I collect board games, must have 60 of them, and lots that I've given away over the years. And I just got a copy of this game over. So it's really cool. It was done in collaboration with a number of editors from Journal of Cell Science. And the object of the game is to develop a healthy cell. And in order to do that, you've got to detoxify the cell, you've got to manufacture ATP, and then you've got to use ATP to drive certain cellular functions. The type of game it is it's called a worker placement game. So each turn, every player has to.
Kevin, I hate to interrupt you. It is 12:10, so I wasn't sure if you wanted to talk about this for the audience or you were just telling me that. But it is 12:10, and I have one question. So your call how you want me to lead this. All right. So hi, everybody. We're starting again. I have one question. If the basal level starts from the same OCAR value in both control and treated cells, can we consider that the amount of cells is similar in all wells? Do we need to normalize them?
I would say yes, because they could be different and just needing different amounts of ATP. So one might need more than another. So if you normalize to cell number, that would tell you. Another metric that might be better for trying to determine whether or not you need to normalize would be to look at the non-mitochondrial respiration, because that's often tightly tied to cell number. Sometimes we coach people about that. I always recommend normalizing data, especially if you're going to try to combine data from different days. But if you want sort of a quick answer as to whether or not you've got the same cells down there, then looking at the non-mitochondrial respiration is usually a giveaway. And you'll see that as we go into some data examples. So I'm going to, is there a follow-up?
Nope, I was about to say, awesome. We are, no more questions as of now, so you can get started. Thanks.
Okay, great. We're going to get into, I'm swapping things around a little bit because finding stuff on our website is fairly quick, but I want to make sure you know where you can find things. I'm a big board game fan, as I had mentioned, so if you like biology-based games, Cytosis is a really cool game where you make ATP to make a happy, healthy cell. So, a lot of fun. All right, so if you go to our website, now I'm using Chrome right now. Chrome works best with our Seahorse Analytics. I'm going to go to www.agilent.com. And this is the Agilent website. There is a lot going on at Agilent. We get involved in a lot of things, very well known for our mass specs. So a lot of information about mass spectrophotometry and environmental and health safety. Lots of things you may be interested beyond just the Seahorse stuff. Seahorse can be found by clicking on products. And then here where it says life science, that's us. And we are part of cell analysis. In fact, Seahorse was its own company, and Agilent acquired Seahorse to form this specific division. Agilent had no cell analysis tools until they acquired Seahorse. So I sometimes like to joke and say Seahorse breathed life into Agilent. So here we have the Seahorse page. On the left is a list of all of the different instruments you can acquire. If you click on these, it'll give you information about those instruments. You can get quotes and so forth. In the center is the consumables. That would be your cartridges and your kits. Click on those and you can purchase those. You can purchase them right off the web. This is where you'll find the software. And this is Al. Al is our chief software designer. And this is Brady, one of his employees. These guys are not lab guys. They're programming jockeys. They just happened to be around the day they wanted to do the photo, so they threw on lab coats and went into the lab. And notice how Al's wearing gloves and Brady's not. So click on Cell Analysis Overview, and this will bring you to a number of different resources for educational purposes. The other items are for ordering materials. You can still order stuff from here, but here's what you got. We've got our information page, all kinds of upcoming webinars, campaigns we're running, various protocols that are specific, like if you're interested in immunotherapy, we've got a whole immunotherapy toolbox. It incorporates many different tools into studying CAR T cells and monoclonal antibodies and so forth, all right here. As we go down, you'll see more information, and right here is our educational resources. Getting Started has a learning center. The learning center has all kinds of information in it. I mentioned the presentation that I just gave you was from the learning center, and there are three presentations in the first 3 sections that I used to make the presentations I just showed you. Gathering materials, you'll notice it's a movie. Preparing your assay is another movie, and you can download these as presentations, and then setting up your assay. Use those 3 to create the presentations that I just took you all through. If we go back to the previous page, we also have a number of videos. There's 2 types, cell analysis videos and cell analysis webinars. What we're doing right now is considered a webinar. If it gets approval, it could be posted right here in these webinars. So you see there's all different webinars broken out by topic, the dates they were broadcast, and the topics are listed here. The authors are here. So if you're interested in T-cell metabolism, activation, and exhaustion by Greg Delgoff, given on October 18th, 2018, click on this and you will be given a link to watch that seminar. Go back. The publication database. I have mentioned this several times so far during the presentation. The publication database has all of the publications that we've been able to collect and has the information about the cell types that were used. My computer has some security feature on it that makes it a little difficult for me to use this. I'm not sure why. It might take me a minute to get started. But over here are your search engines. And here you can simply type in a keyword. A lot of people use this to look for specific cell types. For example, you're running, well, let's see. If I click on cell line, and then here where it says see more, I can expand the list to see all the different cell types that have been used with the Seahorse instrument. And this is where I have some security feature that kind of blocks it. Oh, there it goes. Actually came up. Excellent. And then these are a list of all the different cell types. Click on the one you want or type it in the search here. And it will return for you a list that will link you to PubMed of all those cell types. We'll try this one. So bronchial airway epithelial cells, and this is a list of all of the publications with the cells. And you'll notice that there is a download button here. If I click on that, it will download this as an Excel file. And when I open up the Excel file, and hopefully you can see the Excel file, it shows you the cell type, the assay run, the species, and the seeding density, and if they used any coding for the plates. So this group used these cells at 5 × 10⁵ cells per well, and they were using an XF, well, I did the search by XFe96. So that's how many cells you would likely try to start with. Now, the number of cells you use may be a little bit, you might need to do some empirical optimization because of the way you grow your cells or culture your cells. They may be more or less active than this person's, but this gives you an idea of what other people have done. Plus, it gives you the reference. So who knows, maybe you're interested in silver nanoparticles induced mitochondrial protein oxidation in the lung cells. And back on the web, if I were to click on this, it will take me to PubMed. So I don't have access to the journals from PubMed unless they're free, but a lot of institutes do. I don't know if you're working from home, maybe it's a little bit more difficult. A lot of people use this to get their publications. So that's the cell reference database, one of the more popular places that people visit on our website. And then last is the software. So I'm going to go back. It'd be faster to type it in. Products, cell analysis. You can get this software here, or if you did click on Al, Al's got you covered because he put the software here too. When I click on the software here, it will open up the software page. You want the data analysis software, and there's two versions. There's the Seahorse Analytics, which is going to bring you to a web page where you will register. When you register, you will be given, I think, 20 gigabytes of space in the cloud. Upload your data files. And from there, you can analyze your data files. If you want the PC version of the software, downloadable, installable, you would click on this, you would register, and you get a link to download a hard piece of software to install on your PCs only. The analytics is PC or Mac, and the Wave is just for the PC. So we're going to get started in the Wave software. So, Rule, I switched over to the Wave software. Can you see the software?
Kevin, I can, yeah.
Okay. So this is the Wave software, and I don't know if you can see my screen, but it has an icon that looks like a little water drop. So at the bottom of my page, I've got a little water drop. I would click on that. It would launch the software. And this is the opening screen. You notice how it defaults to templates. This is where we would create an XF template for running an assay. The Seahorse Analytics, the cloud-based software, does not have this feature, so you can't create a template using that software. That software is just for data analysis. This software, do the template on your laptop, or you can do it right on the instrument because it's the same software on the instrument. To create a template, you could use one of the pre-made templates, which is the icon here. If you wanted to run, say, the cell stress test, you could click on this, and it will pre-populate all of the fields with information relative to that assay. The fields look like this. I'm going to start with a blank, and I'm going to click Open File. The first screen is the group definition screen. On the left-hand side are categories for our groups. For example, perhaps I'm running two assays or two different injection strategies. On the same plate, I want to do my glycolytic rate, and I want to do my cell stress. Notice there's a little 2 here. That means I've got 2 groups so far. And then I can define what's in the port. So for the GRA assay, the first compound in this case, I recommend would be vehicle, no concentration necessary. Port B, I would add a compound, and this is going to be my rotenone mixed with antimycin. The final concentration will be 0.5. And then the third port will be 2-deoxyglucose. Final concentration of, I think, 25. And I would do the same for the cell stress test. The drugs are different. For example, port A would be oligomycin. And I would repeat that process for ports B and C. Now, it's important to note, this is just information. It's going to be in your data when you get it back, but it has no bearing on what the software or hardware does. We will get to that on another screen. This is a type of digital notebooking. It's great because it lets you know all of the variables that you threw at your cell and what those variables entail. If you have any pre-treatments, you could add that here. Perhaps I've got a wild type and a knockout. And notice how there's a big description here. These could be C57, black 6, 3-week-old females with vector. And then pre-treatment 2, knockout, CKO protocol. And notice how there's a 2 here. And then I can enter in my assay media and the cells that are being run. Maybe these are, I don't know, T cells, thymocytes. The seeding density, when they were prepared, and so forth. So right now we've got a pretty simple design, 2 by 2 by 1 by 1. One thing about the assay media is there is an option to use our media. Any media that has a pH associated with it will automatically give you buffer capacity. So when you use these media, it will recalculate your data to get that proton efflux rate data. So I always recommend using this so later on you have that information. Now the buffer factor is the buffer capacity adjusted for the volume that's measured in the instrument. And this is something that's determined by Seahorse. So this media may have a different buffer factor than your own media. If you make up your own media, we have a protocol to calculate the buffer factor, and there's a spot to enter that information in when you get your data back to recalculate your proton data to get accurate proton efflux rate data. I can go into that in more detail later, but for now, just want you to be aware of it. Once I've created my groups, I click the Generate Group button. What's going to happen is the software is going to multiply these together, color code them, and put them over here for me. These are all my groups now. Now I'm going to go to my plate map, and I'm going to assign these groups out. Now I did a template for an XF24. Notice there are 4 wells. Remember I said before you want 4 negative control wells. These are them. They're pre-populated. It's a group called background. If for some reason you want to move them around, you can. But we always recommend having 4, okay? Whether you're doing the XF, well, 4 for the XF24 and 4 for the XF96, 2 for the XFP. And they would be on the ends, well A and well H. For the XF96, they would be in the corners. Okay? No cells go in these wells, only your assay media. Now I would click on the group and I would add it to the wells that make up that group. You can click on a letter, you can click on a number, or you can left click and drag. However you want to set this up. So these are my statistical groupings. I recommend running 4 replicates per group, minimum. For the XF96, when you first run your assays for the first time, I recommend 6. This helps to improve your error. And if you find that your error is really small, you can drop it down to 4. Sort of a poor man's power analysis. After I've set up my plate map, I'm going to go on to the protocol. This is the most important part of this programming. Everything else could be done later. There are modify tools in your data analysis that allows you to change your groups, change the names of the groups, add buffer capacity, and so forth. This part controls the hardware. I say it's like programming Robbie the robot. To instruct the instrument when to measure, when to inject, how many measurements to do, and so forth. On the left-hand side here is some housekeeping. You need to calibrate. This is just a reminder. And then after you calibrate, there's an equilibrate step. Calibration is done with the cartridge sitting on top of the utility plate in the calibrant. You cannot calibrate the instrument in water, or you cannot calibrate the instrument in cells. So you start with the utility plate and the cartridge on top of it. That goes in the instrument first. The calibration takes 10 to 20 minutes. It's simply measuring a buffer and calculating a lookup table to tell you how much fluorescence is equal to a pH of 7.4 or how much fluorescence is equal to an oxygen of 20%. That requires the calibrant and the utility plate and the cartridge. When that 20-minute period is over, the software will stop the instrument and an announcement will come up saying the calibration is complete. Please click continue to continue the assay, or please insert your cell plate now. I forget the exact message, but the instrument will stop and wait for you. When you get to the instrument, click continue. The tray will come out and the utility plate will be sitting there. The cartridge will be hanging from the inside of the instrument. Take the utility plate with the calibrant out and replace it with the cell plate and click continue. The cell plate goes in and then the instrument will start making measurements within about 6 minutes. When you're programming, it automatically gives you a baseline. If I click on this little expand button here, it shows me what a baseline measurement is. In this case, it's going to repeat these steps 3 times. It's going to mix for 3 minutes, wait for 2 minutes, measure for 3 minutes. That is standard. That does not need to be changed.
You're breaking. Try it one more time. No worries.
Where did I leave off?
I didn't hear the last sentence. If you could just repeat that.
So this part here indicates that you're repeating these steps 3 times. You're mixing for 3 minutes, waiting for 2 minutes, and measuring for 3 minutes. Now on the XFe and the XFe96, the wait is not necessary. So this would be 0 for those programs. If for some reason you go back to your labs and you go to run an assay and you remember 323 because that's what I showed you, and you're running an XF96, it harms nothing. It is fine to do that. Some people who migrated from a 24 to a 96 prefer it because they don't want an extra variable. However, if you remember, you do not need to put a wait command in the XFe96 or the XFe. In fact, when you go to program them, the default will have a 0 there anyway. The other numbers will be the same. So right now we have 3 baseline measurements, and each measurement will take 8 minutes. If you do the math, it's 8 times 3 is 24. The equilibrate command is just warming your plate with the cells on it for 12 minutes. So that adds up to 36. I wouldn't touch this unless you're running isolated mitochondria, then turn it off. It basically just skips that step. So you start measuring more quickly, which you'll need to do if you've got something that's not going to last for very long. All right, now what do we want to do? We want to inject. So we click the inject command, and the software will automatically select the first available port and append 3 more measurements to it. Perfect. That's what we want. If for some reason we want more cycles or fewer cycles, we can do that. When would you change this? Most times you wouldn't, but let's say you're injecting a drug and you have no idea of the kinetics and you want to measure for an hour. Well, each measurement's 8 minutes long, so I would put 6 or 7, well, 7 or 8 measurements. Then you'll get a measurement every 8 minutes over an hour-long period. We inject our next drug and then our final drug, and we've completed the process. If we want, we can name these. If you remember, this is 2 assays in 1. So this would be vehicle or oligo. And then this one would be rotenone or FCCP. And then this one would be 2DG or rotenone. Click on the Run Assay button. If I want to, I can save this as a template. And if I'm ready to go, I can start running the assay. If you do this on your laptops and you save it as a template, it will show up back on the home page as one of these green files. It doesn't physically save it. It puts it into the template screen. If I want a physical copy of this, click on it, and down here is an export button. Export it to a thumb drive. For those of you using an XFe, you can do this as well. Make sure that the template goes in the root directory of your thumb drive. Don't put it in a folder. When you go to the XFe and plug your thumb drive, the XFe only reads the root. So it won't tell you what folder structure you have. So that's how you do it with the XFe. At this point, I'm going to switch over to doing data analysis. So do you guys have any questions about programming?
Kevin, I have a few questions that came in. So the first one is, could you please explain the injection strategies again? Is this cell treatment for the assay and not the ports A to D assignment?
So, whoops, wrong button. All right, so we go back to the group definitions. The injection strategies refer to what you put in the ports. What's your strategy for injection? Strategy for an injection could be anything from a cell mito stress test to a drug titration. It basically indicates that the ports are getting different compounds. A pretreatment would mean that your cells are being treated differently prior to the start of the assay. They're cultured in different media. They come from different sources. They're wild type versus knockout. Assay media could be used the same way. Maybe your media is high glucose versus low glucose. These different categories basically represent the variables that you could throw at your assay to create all of your statistical conditions. There are many, many different injection strategies. I suppose there's an infinite number of injection strategies. A lot of times people use the injection strategy category simply to indicate they're running a cell-minded stress test or a glycolytic rate assay or one of the pre-made assays that go with the Seahorse kit.
Okay, thank you. I have another question that has a follow-up that said it was addressed, but because it's around the HS Mini, perhaps it's worth explaining to the audience what the HS Mini is. So the question is, can you create a template on a computer and transfer it to the HS Mini, or does the template need to be created to the touch screen of the HS Mini? So perhaps, Kevin, you could explain what the HS Mini is and then answer that question for the audience.
How much do you want to go into the history rule or just talk specifically about the HS Mini?
No, specifically, what is, because it's a new product and everybody might not be aware of it, and the question is around that specific product.
So the HS Mini was developed by our engineers to work with material that provided very few cells. We've talked about cell density. You saw in the Excel spreadsheet when I pulled down the bronchial epithelial cells, they ran 50,000 cells. I mentioned anywhere from 7,500 to 400,000 cells. So there is a cell minimum requirement for this instrument. I know folks that do cell sorting, work with T regulatory cells, and they only get like 200,000 cells per draw. Now, if you only get 200,000 T regulatory cells, that's enough for one, maybe two wells in an XFP or an XFe96. So you can't really run very many conditions, and it won't work at all in a 24. You just need too many cells. So the HS Mini was developed to allow you to use fourfold fewer cells. So those 200,000 T-regs you just pulled up, for an HS Mini, that's 4 wells. So you'd be able to run a couple of conditions there, maybe more, because it does have a high sensitivity. So you use fewer material in the HS Mini, and the interface for that is very similar to the, you can use this software to program it offline. Online, it has its own touch screen, which is really slick. I don't have any way of showing it to you because I don't have an HS Mini here. There were some slides, but I don't have them accessible, that show you all the different screens, but it's very intuitive. You click start, then it asks you to name your groups, you name your groups, then it asks you where you want to place your groups, you put your groups there, and then it just asks you how many injections you want to do, and you're done. And it's just like 4 touch screens that you go through on that. If you want more detail, you would use the Wave software like I just described.
Awesome. Thank you, Kevin. And again, I put a follow-up note in case the person who asked this question needs a follow-up for the online piece of it. Another question, what is the reason for needing the wait time on the 24 but not on the 96 platform? And can the XFe24 assays be run with 0 wait time?
I forgot to mention that. The XFe24 runs in a larger volume than the E96. During the mixing phase, you are plunging up and down, which caused some thermal instability. So the engineers put a wait command in to maintain the temperature, bring the temperature back to stable. In the XFe96, the volume is smaller and the wells heat up faster, so the wait is not necessary. Can you run a 24 assay without the wait? Yes, but you may see higher than normal variability. Not crazy variability. Like, we're not talking about going from a 5% CV to a 20% CV. We're talking about going from a 5% CV to maybe a 6 or a 7% CV.
Okay, thank you, Kevin.
Okay, good.
Yeah, sorry, I was asking myself the question. My apologies, I was muted. So is the wait period before the first measurement necessary? And that's the last question we have for now.
It's almost a philosophical question. The wait command then is again for stability. Temperature changes will cause variability. everything is warm, if your material is well warmed, you're using thermal trays to carry things around, your media is warm, then that wait command becomes unnecessary. If things cool off, the wait command is there as a safety feature to help things warm back up again. And we turn it off on purpose when we run isolated mitochondria because they've got about a 3-hour window in which to use. So we want to get measuring right away. And temperature is less of an issue when you're working with an organelle than with a whole cell. So the answer to your question is I would leave it on, but you could turn it off if you felt that everything was adequately warmed.
Okay, thank you, Kevin. I have no more questions for now.
Right, so let's talk about data analysis. So from the results screen, you can open up data files. You simply click on browse and you can browse to whatever folder where your data happens to be. And right now I am using the Seahorse Wave software. So we're going to open up this file here. And it will open it up, which it already did, right here. So when you open up the file for the first time, this is the screen you're going to get. It's called the Quick View screen. It shows you the OCAR data, the ECAR data, and the energy map here. The energy map is your histogram and is linked to one time point, not a kinetic graph. Right now it's displaying the very first measurements data. You can cycle it around by using this dropdown. You want to see what happens after I add oligomycin? Well, that's what happens. This is generally used just as a quick view to let you know what assay worked and some basic information. To drill deeper, you want to click on Add and create another view that we call an overview. The overview will show you one variable at a time. Right now, it's showing me the oxygen consumption data. So this is the ATP rate assay. We've injected oligomycin, and then we followed it up with rotenone and antimycin. After the oligomycin is injected, you see a decrease, the rotenone, a further decrease. So it looks like the assay worked. Down here is a legend that shows that I've got a control cell, treatment 1, LPS, interleukin 4, and treatment 2. So these were macrophages that were called 48 hours in the presence of these different treatments or in vehicle only. And you can see that the LPS has done something, the interleukin 4 has done something. And I mentioned before that if your basal rates are right on top of each other, I would still recommend normalizing the data and that the non-mitochondrial respiration might be more informative as to cell number. Over here, you see the non-mitochondrial respiration, and the LPS treated group appears to be higher. That likely means that the LPS treated group grew faster, stimulate the macrophages, and they divide perhaps. The group that I spoke with said that they don't really divide under these conditions. So this then would be likely reactive oxygen species. The cells have become activated and they've started to produce more ROS. You can test either one using other tools. That's sort of what the data is telling me so far. Notice how there's no information yet about ATP. I can switch my view from OCAR to ECAR, and there's my ECAR data. When they're stimulated with LPS, way different. So the assay appears to have worked. Interleukin 4 has done something as well compared to the other groups. So it looks like a nice assay, but I still have no ATP data. To get the ATP data, there is a button here called Export, which will allow me to export the data to any one of these particular tools. I would choose the assay that I ran, which in this case is the ATP rate assay. Click on that, and it will ask me where I'd like to save my data. Notice how it's saving the data as an Excel file. So click Save, and the data is now saved as an Excel file. Now I just need to go and open it up. So I'm navigating, although you can't see, over to the sample data page, and there's the file. I'm opening it up. And now you should see Excel. And macros have been disabled. All right, so enable the macros. Now a screen pops up that says, which groups would you like to compare? So I will select them all and now update the summary. What it's going to do is count the rates of ATP produced from both pathways. So it redraws the graph, and now I see the total amount of ATP produced from each condition. My control condition, PS stimulated condition, interleukin-4 condition. It also shows me in red the amount of ATP produced from glycolysis, and in blue the amount produced from mitochondria. When I stim the cells with LPS, you can see that a greater portion becomes glycolytic. You also see an increase in oxygen too. With interleukin-4, there's not a big change in glycolysis, but there is a big change in oxidative phosphorylation. This is a hallmark of M0, M1, and M2 macrophages, which I think you may have an interest in yourselves, but I highly anticipate a lot more work is going to be done on these cell types because they're the ones that are responsible for pro and anti-inflammation. They're responsible for tissue repair and in some cases tissue damage that leads to the upper respiratory damage we see in patients with COVID-19 that wind up in the ICUs due to upper respiratory distress and cytokine storm, which is oftentimes due to these cells. So interleukin-4 can reverse that. And I learned yesterday from one of my colleagues that you can't take an M1 cell and make it to an M2 cell very easily. So a lot of good research for all that. Down at the bottom of the page, you see a measurement sheet that shows you your group statistics data, assay parameters per well, which is your raw data. This is data that you might export to Prism to do statistics. And then project information lets us know about the coefficients for the cartridge, the groups that were included, the buffer factor, and so forth. So that's all done with, back in the Wave software, I want to go over a few other features that may be informative and helpful for you. So up here is a normalization button. If I click on it, you'll see a table pop up and it's blank. This is where I would enter in cell number per well, protein content per well, whatever it is I've normalized to, and the software will automatically divide each rate by that number. They give you picomoles per minute per cell. I don't have any information for that, so I'm going to click cancel. Next to normalize is the modify button. This allows me to change the names of the groups or even where they're located on the plate. Simply click a group and assign it to its new location. The summary button. When I click the summary, bang, nothing happens. So what does the summary button do? There is a summary page here where you see your views. If I click on the summary page, it shows me a summary of my data. And when I click that summary button, I basically copied that screen and pasted it into the summary. Now you can see I've already put some information in the summary. Up here you see the date that the assay was run, the time. Tessa was the primary user, and I was just joking around, so she was a little nervous when I used the summary button, but that's what the summary does. These buttons here are linked to the information you entered in during your programming. If I click on the protocol button, it shows me the mix, weight, and measure times, the number of measurements that were used for this assay. The group button shows me the groups that were used for the assay, how we named them, and ironically, it didn't save the names that I changed them to, so she actually did an experiment on this. And so forth. You can export the data as a PDF or as a Word document, and then use that for your grants or for your publications and whatnot. Now I want to get out of this screen, so I go back to my overview screen. And from here, if I want to, I can take a closer look at the data. The LPS group seems to have a bit of variability. There's a couple ways to examine that. There's a group button that allows me to switch from group to well. And you can see there's quite of a spread there. So I could go and look at those wells to see if they were seeded unevenly. If this has something wrong with it, I could double click on it to remove it. You can see that it is grayed out now in the plate map and it's no longer being the averages here. There's also a group and well button here that allows me to take a look at the wells individual rates at the time point indicated here. So again, I can cycle through the different time points, look at the rotenone, and you can see they all look pretty nice there. And then I can go back to looking at group. Now this button here allows me to express the data as a rate or a level. You want to know the level of pH or level of oxygen? Switch to level mode. When I switch to level mode, you'll see that the assay was run pretty nicely right at 7.4 throughout the duration of the assay. If I switch over to the OCAR data, you'll see that the OCAR data is sitting right in ambient and it's coming up and down throughout the duration of the assay. So no air bubbles, oxygen, and pH look fantastic. So I just wanted to give you sort of a feel for the WAVE software. This is how it works. Next week, when I do the deep dive data analysis, I'll be using this tool a lot more. So you'll get to see more functions within the WAVE software. Now, if you happen to use a Mac, we have, no thanks, we have Seahorse Analytics software. So when you do the Seahorse Analytics software, you'll get a link to go to the Seahorse Analytics page. So you can see seahorseanalytics.com as a dashboard. You'll type in your username and password and you'll get to the dashboard. To upload data files, you would use this button here. So if I wanted to upload that data file that I just showed you, click on the cloud button. Oops, maybe I need to log out. And it would open up the ability to upload your data. So let's see if I needed to log back in. Yeah, I needed to log back in. Everybody cover your eyes. Just kidding. Whoops. Oh no. It looks like I've been locked out permanently because I left it on for too long. I got a confirmation email. All right, it looks like I got locked out because I had left this running, hoping I could quickly switch to it. So I won't be able to show this right now. Let me try again. Looks like I've been locked out. I'm sorry, I can't do the Seahorse Analytics. I was able to show you the dashboard, and then when I tried to upload a data file, I got locked out. So I need to a new password. I did not know this was going to happen, but it looks like the caution here is don't leave the Seahorse Analytics open for longer than an hour without interacting with it. I'll mention this product manager. If you tune in next week, I will use the Seahorse Analytics then. What I really wanted to show you how quickly you could get those graphs. So in the WAVE software, I had to export the data to Excel, then open it up. In the analytics software, I just need to click on the graph I want and it appears. So I'm sorry I can't show you that the last 10 minutes. Oh, you know what? Maybe
Kevin,
I can.
I do have a bunch of questions that we can address in the next 5 minutes, and then just we can make sure that this is addressed first thing in next week's meeting.
Okay, I think I realized what I did. I used the wrong account. There we go, I'm in.
Oh, there you go, perfect.
Do you want to answer the questions or do you want me to show Seahorse Analytics? This was my fault. This
You
was not
tell me. You tell me. You're leading this event.
Let me quickly show you this. So you would click on the cloud and then this would ask you to load up a data file. I wanted to show you this data here. This is from E. coli. So I promised I would show some infectious disease data, and I haven't shown a heck of a lot. So this is data from a demo, from a training we did, and it's E. coli. So when I open it up, I get this screen here, and it shows me all of the data plus the different categories we use. This optimization assay, this is the first time I ever ran E. coli. So we did a lot of things here. We treated the plate with and without poly-D-lysine, different densities of the cells. And what we have here is called the kinetic graph. I can expand this, and there's all different types of views. I can click here to get all the different standard graphs. We can do a kinetic graph, bar chart, energy map. Energy map might be neat. Click the add widget, and then it just appears here. we've got our plate map on one side, all the different conditions here, and this guy kind of stands out. It was a plate that was coated with poly-D-lysine, and we had, I think we had 8 million bacteria on the plate, and we got really nice rates with this guy. We did further optimization, looking at things like glucose, but I just wanted to show you the overall layout. If I go back and we go home, I had mentioned how quickly you can get some of these graphs. Now this E. coli data wasn't one of the kits. We just ran a base assay with pyruvate to see if we could get anything. And that's what you're looking at. Notice how it's pyruvate and FCCP. I'm going to go home and then upload a data file, choose a file. And this was the assay that I just showed you on the Wave software. So we'll import it. Click on it. And I really just want to see the data. Make sure that it knows that oligomycin was injected through port 1. And I can get these data. Click Add View, and it's all going to come back for me, just the ATP data. Whoops, forgot. Oh, I forgot that. Darn, I thought that would carry over. All right, it's asking.
Kevin, we have a question that's relevant to what you're doing here. Someone didn't catch what the difference is between Wave and Seahorse Analytics as far as analysis is concerned. Can you address this question?
They're essentially the same. The big difference is that the analytics is cloud-based, so you don't need any software. You just need a browser. Sorry, I thought I had this information in there already.
No worries.
So
I do have more questions.
If you double click on it, it opens it up just like the Wave software. So now you're going to see a screen that looks just like the Wave software. What other questions are there?
Okay, so we have 4 minutes. I do have 5 questions. So you think we could go through all of them or just a couple and we could follow up with the rest? Do you have anything else to show?
No, I forgot to put the buffer factor into this data file, so I can't actually calculate the ATP data, but the steps I went through is how you do it. I just forgot that one variable.
Yeah, so we did get that question. How should the buffer factor and the ATP rate assay be determined?
It's predetermined by our media, and I forget where the field is to enter it in on the analytics. But if we go back to the Wave software, what it's asking for is for me to go into the software and modify the assay media. So click on assay media, and right here where it says configure, I forgot to configure it. The background wells don't have any buffer factor in them by accident. I just forgot to click these boxes. I'm not sure why the engineers designed it this way. When you use our media and you run these assays, the buffer factor is determined and for each of the wells and is automatically entered into your variable wells, but not into the blank wells. The reason I think the engineers did it this way is because if you're running 2 different media conditions, you'd get 2 different buffer capacities. So you'd enter them in differently because we recommend entering them into their own background wells. I think that's why it's like this. At any rate, I just forgot to check these. If I now save it, the background now has the buffer factor. Then if I were to save this data again, oops, I forgot to apply. Then I could upload it into the analytics and it would be fine. Now, this same screen is in the analytics software somewhere. I just can't remember where right now. I usually do this in the Wave software, but it's 6 of one, half a dozen of another. Once you get familiar with the software, you know where all the buttons are, it's fine. Next question.
All right. Next question is there a library of examples or demo experiments that I could use to explore the software?
There is a little bit on our website. If you go back to Agilent.com and spell it right. In the Learning Center, so products, cell analysis overview, click on Getting Started and then Learning Center. There are some parts of this, like Analyze Your Results, that walk you through some of the steps of the software relative to that instrument. So the XFP, the HS Mini, which I don't think has been added yet, and the XFe96 will show you screens and details. From the software. Additionally, a lot of people miss this. When you register for the software, even if you're going to use the analytics software, when you register for the Wave software, you will get a PDF to download that is a user manual. So a lot of people miss that there's actually a Wave user manual. But I said, even if you're using analytics, this could be helpful because the two softwares are very similar. So there's information in the documentation for the Wave software that will be very similar to the analytics software.
All right, thank you, Kevin. We are at the top of the hour, so unfortunately, we cannot take more questions. However, if there are any remaining questions that were not addressed here, please email them to myself or Kevin. You have both of our emails. They were chatted in earlier. If you want to scroll up while I'm concluding. Also, as a reminder, we will follow up with an email to address all the questions that were answered today. Wanted to thank you everyone for your presence and participation. I'm concluding this meeting by announcing that this meeting has now ended. And again, thank you so much for your participation. Thank you and stay safe. Appreciate all the efforts today. Kevin, thank you as well. You did a great job. Appreciate it.
Have a good day, everybody.
Bye-bye, everyone.
Hello. Hello. You there? Thanks. Oops. All right, some people are still on the meeting.
How to quit?